Mm. Okay, good morning everyone. So now it's already 8 o'clock. Okay, okay. So now it's already 8 o'clock. We will start our revision now. Okay. So yesterday we already complete 4 chapters. So today we will complete the remaining 3 chapters. Okay. So now it's already 8 o'clock. We will start our revision now. Okay. So I think I already closed the background music. Okay. Okay, so now 8 o'clock we will start our chapter 11, chapter homeostasis. So in this chapter, as you guys know, we will have 4 topics, right? First topic is about importance of homeostasis, and then second topic is about liver structure, liver function. Third topic is about osmoregulation in mammalian, and fourth topic, like final topic, is about the osmoregulation in plants. Okay, so we will proceed to the 4 topics, right? First topic is about okay. So we will proceed to the first topic first. Okay, so about the importance of homeostasis. Okay, so you need to know the definition of homeostasis. So what is homeostasis? So actually, before you learn this chapter, in previous chapter, actually you already done lah. All the mechanism that include the process to maintain the constant internal environment by the body of an organism. So it is actually is the process or the ways to maintain that the environment or the condition inside the body is always maintained in constant or suitable for body to survive. So before you enter this chapter, actually in previous chapter you already learned about some mechanism about homeostasis already, like your first chapter, chapter 7, cassette chain breathing mechanism is one type of homeostasis, and your regulation of heartbeat, that second chapter, is also one type of homeostasis too. And then your fourth chapter, chapter 10, the last chapter, the mechanism like the urine penetration, also one type of homeostasis, and then one type of homeostasis too. So actually you already learn lah. Although you not enter this chapter, but this kind of mechanism is also considered as one type of homeostasis. So actually this is the main component of homeostasis. So normally you will get the firstly your receptor, your sensory receptor. Your sensory receptor, like the sensory neuron, any sensory receptor, you will receive the stimulus first. Maybe there's some change, like your blood pressure change, your blood pH change, or your temperature change. All that, when they are detect some change, it will detected by the receptor, and then this receptor will be detect the stimulus, and then it will go to the it will change. It will be compared to the set point. The value will maintain for a particular variable or optimum level at the system which operate. Okay, and then it will go to the effector like your muscle or your glands to secrete your muscle to either contract or relax, or your gland starting to secrete hormone to respond it, so that you will have a respond to have a activity to return your set point. return your value into your set point. So these are the components that is very important in homeostasis. Your set point. Return your value into your set point. So these are the components that is very important. Okay, next is about the mechanism in homeostasis. So as we know that we have two type of mechanism, right? Positive feedback mechanism and also negative feedback mechanism. In human, normally we will proceed to negative feedback mechanism, meaning that when there is detect a change, the mechanism will be reduce the initial stimulus. We want to return the abnormal particular condition back to the set point by the effector. But for the positive feedback is to amplify the stimulus. So normally one of the example is during patriation that you learned in last chapter. Only in human normally one of the example mechanism, but in negative feedback mechanism almost all So like thermal regulation or small regulation, but glucose level later we will learn it is one. It's one of the example of negative feedback, and also your breathing mechanism that you learn in chapter 7 also one of the example of negative feedback mechanism. Okay, next we go to the importance of homeostasis. So why homeostasis is very important? Because it is the dynamic equilibrium process to interplay between external factor and internal control mechanism. So it will produce the steady state of your internal environment to make your internal environment is always constant, even your external environment is changed significantly. Okay, and then there are the physiological activity response by stimuli are not instantaneous, meaning that your internal mechanism will not change the environment immediately. Like when your environment temperature is increasing very high, so that in homeostasis they will maintain your temperature by like 37 degrees Celsius As a result, homeostasis can moderate the change of internal environment only, not elimination the internal environment. And then it enhanced by some organism like hormone or your effector in body to reduce fluctuation. It also allow biological system to function efficiently and also can live in wider range of habitat. So like human, you can survive like even though in North Pole negative temperature, you also can survive like in desert up to 50 degrees also you also can survive like that. And also it allow enzymatic variation and cellular activity go to the normal in the body. Okay, next is about the thermoregulation. So this is the regulate your temperature. So this is a process by which your animal maintain their body temperature within a normal range. Okay. In terms of thermoregulation, we have two types of organism, endothermic and also ectothermic. So in our syllabus, we will focus more on endothermic Ectothermic is not our in our syllabus. Okay, endothermic is the organism like human usually warmed by heat generated by metabolism of the cell. But the ectothermic organism like snake, the organism gain the heat from the external sources. Meaning that the temperature will be changed based on the environment. But for endothermic, the internal temperature is remaining constant. But ectothermic, the body temperature will be changed based on the environment. Okay, so I already mentioned endotherm is in our syllabus. Endothermic is our syllabus, ectothermic is not in our syllabus. So we will more focus on endotherm. So there are the 6 ways of thermoregulation in endotherm: insulation, blood vessel contract or relax, counter current exchange, evaporation, cooling effect, thermogenesis, behavioral response. So these are all the thermoregulation that they will be done by endothermic organism like human one of the example is human. Okay, about the insulation. So what is the meaning of insulation? So insulation actually is one type of process to reduce the flow of heat between the animal body and its environment. Okay, so it reduces the flow of heat. of heat. So normally insulator is normally found at the body surface lah, like hair, your feather, and even though for those organisms that have the adipose tissue. Okay, so insulation normally will secrete oily substance that will repel water, protecting the insulating capacity of the feather or fur. And then animal can adjust their insulating layer to further regulate the body temperature. So normally during hot environment, so normally insulation play big role during cold environment. Because it need to reduce flow of heat, right? Already mentioned, reduce flow of heat. So when we want to reduce flow of heat in cold environment only, right? In hot environment we don't need reduce flow of heat. So only during cold environment your organism fur or feather will rise. So you see that in human, our skin will also become will be rise, right? Our skin hair will also rising, and then the action create a thicker layer of air, thereby increasing the effectiveness of the insulation. So the organism that lack of fur like human must rely primarily fat of insulation. So in marine organisms, so they swim in water colder than their body core. So many species spend at least part of the year in nearly freezing polar sea. So that is why that in marine organism the adipose tissue is very thick compared to human. In marine organism their body is containing many fat one. Okay, so why they want to like this? They need to transfer of heat to water occur more rapidly than heat transfer to air. So survival under this condition is made possible by an evolutionary adaptation called blubber, a very thick layer of insulating fat just under the skin. So that is why that I already mentioned why that the marine organism contain many fat, because it need to transfer of heat to water, and then they need to maintain the body core temperature without requiring much more energy from food than mammal. Okay, so this is about the insulation. So normally insulation is due to the either presence of body surface or hair and feather or adipose tissue. Another one is about the blood vessel contract and relax. So normally this one is including the blood circulatory system. So the blood circulatory system provide a major role heat flow between the interior and exterior. So normally this one is about your surface blood, your skin surface blood vessel, because this one is play the role about the heat flow between interior and exterior. So when the blood, when your blood is already flow onto the surface of the skin, heat flow will be done. Okay, so in response to the change in temperature, many animals will alter the amount of blood flowing between their body core and their skin. So normally there are two conditions, either the blood vessel is contract or relaxed. So normally in hot condition your muscle will be relaxed because it want to widening your blood vessel. So why it want to widening your blood vessel? Because it need to increase the blood flow in the skin, because you need to transfer heat from your interior body to the exterior of the body, because it is too hot. Your body cannot maintain your body temperature in too hot environment. So you need to transfer the heat from your body inside to outside. Okay, during cold condition, you need to reduce the heat. You need to maintain your heat in your body, so you cannot transfer the heat to the outside already. So you will have the vasoconstriction. Your blood vessel muscle will start to contract. So when it is contract, it already reduce blood flow. So the heat transfer will be decreased, and their diameter of blood vessel also will be decreased. Okay, next is about the counter current exchange. So this one is also like the blood circulatory system, but it is the heat exchange between the artery and vein inside our body. For this, it is normally occur in the surface of the body, the blood vessel that be presented in the surface of the body. But for this is about inside the body. So meaning that this is actually the counter current exchange effect, meaning that in our artery and the vein, so normally the artery and the vein is flow in the opposite direction, right? Opposite direction. So normally artery is flow from the heart into all part of body, and the vein is flow from the tissue payer to into our heart, So they are totally in the opposite direction. So normally artery will carry warm blood. Is carrying warm blood. The keyword is carrying warm blood, and then our vein is carrying cool blood, the cold blood. So that since they are arranging in the opposite direction, the transfer of heat can be occur. So near the end of the leg, where your arterial blood has been cooled to far below your core temperature, the arteries can still so this arrangement facilitate the transfer from artery to vein, heat transfer from artery to vein along the entire length of the blood vessel. So since they are flow in the opposite direction, so the arteries can transfer the heat from the artery to the vein directly. So the blood in the vein continue to absorb the heat as it passes warmer and warmer blood traveling in the opposite direction. So it is what we call as counter current. So it already counter the effect of it. And as the blood in the vein approach the center of the body, it is almost as one as the body core. It will minimizing the heat loss that result from the supplying body to body part increase immersed in cold water. So this process is to prevent the heat loss. So if no this process, meaning that your vein temperature, the blood in the vein temperature is very cold, so that when it reach the center of the body, actually the heat transfer will be occur rapidly in your skin. So if this process happening, so that your temperature normally is already equal to the body core temperature, so that the heat transfer done in the skin will be minimized. Okay, so this is about the explanation. Okay, so most importantly, heat is transferred along the entire length, minimizing the effect of heat exchange. Okay, so normally this one is very important in marine organism, and then they use this mechanism to keep the main swimming muscle warm, enable them to swim, and also some of the endotherm insect will also remain high temperature in their thorax, so that they can where flight muscle are located. Okay, next is about the evaporation cooling effect. So this one I think everyone should be know, because this one is one of the water properties that you already learned in chapter 1, semester 1. So normally if you know that in semester 1, one of the property of water is higher heat of vaporization, right? Heat of vaporization property. So that if environmental is above our body temperature, evaporation can keep body temperature from rising. So it is to maintain the body temperature not increase very well. So that is why in very hot condition you are sweating. You will be sweating. So that your property contribute to evaporative cooling effect, where water molecule. This property will contribute the cooling effect, where water molecule with the greatest kinetic energy live as gas, making the remaining water cooler. So I think this one everyone should know that when you are sweating, when you have the water on your skin, so normally your water will be evaporated, leaving as a gas, but the remaining liquid will be maintained inside on your body surface. or the user face. So this remaining liquid will act as the cooler lah. So the water will only absorb the considerable heat that it is evaporate. This heat is carried away from the skin and the respiratory surface with water vapor. So evaporation of sweat from skin dissipate body heat and help to prevent overheating on a hot day. And also some birds have a pouch visually supplied with blood vessel in the floor of the mouth. Fluttering the pouch increase evaporation. Okay, another one is about the thermogenesis. So normally this one is only occur in cold condition only, because it need to maintain your body temperature higher than environment. So normally this one is only maintained inside during cold condition only. In hot condition, this process will not be occur. So normally it need to thermogenesis need to produce heat production to prevent the heat loss. And then your thermogenesis normally can be increase your rate of thermogenesis by muscle activity, like you moving or you shivering, like you do exercise, then you can warm up your body like that. They are like the small organism like bird. Shivering can make them remain reactive and hold their body temperature nearly constant. And therefore the insect, endotherm insect, so normally they will shivering their wing before they fly. So they are the chemical reaction occur, and then resulting the cellular respiration can accelerate in warm up flight motor and ability. They can fly even in cold air. Now in mammalian like us, so they are the endocrine signal in response to cold cause the mitochondria to increase their metabolic activity and produce heat instead of ATP by non-shivering thermogenesis. So mitochondria also not only can produce ATP, it also can produce heat only. So for this time, your metabolic activity for mitochondria just produce heat, not ATP. So this process we are called as non-shivering thermogenesis. And also some mammalia also have a tissue called brown fat in their neck and between their shoulder that is specialized for rapid heat production. Okay, last one is about behavioral response. So normally behavioral response is very directly respond lah. What respond when you were cold. So normally when you are exposed to cold environment, you need to seek, you will try to find the warm places, and then you need to orientate themselves toward heat source, and then expanding the portion of the body surface to expose to the heat source. And when you are in the hot condition, you need to bath, awak akan mandi kan, and then you need to move to the cool area like aircon area. You need to turn on aircon or switch on your fan like that, and then you need to turn in another direction to minimizing their absorption of heat from the sun. Okay, now this previously we are learned about the thermal regulation in endotherm. So now we will more focus on human skin. Okay. Human skin, the thermal regulation, the opening that occur in human. So I think the structure of skin I don't need to mention more lah. So normally structure of skin consists of 3 layers, right? Epidermis layer, dermis layer, and also subcutaneous layer. So subcutaneous layer is the layer that contains adipose tissue lah. And then we know that we have the sweat gland on our skin, and then we have the hair on our skin, and also we have our cold receptor and heat receptor in our skin like that. Okay, so normally this is the overall process about the negative feedback mechanism of thermal regulation opening in human. So normally this is our normal temperature. So if you see the upside one, so normally when your body temperature is increasing in hot condition, so normally your thermal receptor in your brain will starting to stimulate, and then it will send the impulse into your effector. So normally your blood vessel was starting to dilate because it need to increase the blood flow, vasodilation. You need to transfer your blood flow into your skin surface to increase the heat transfer, and also your sweat gland was starting to secrete the sweat like that. In your cold temperature, your effector, like your skeletal muscle, will rapidly contract, causing shivering, and then your blood vessel will be contract also. Okay, so this is the regulation explanation in hot day. So normally heat load central within your anterior abdominal now will be activated, and they will send the impulse to the effector. And then your nerve signal will transfer to the muscle of the blood vessel wall, causing the muscle to relax, or vasodilation. So vasodilation I already mentioned that, so I don't need to explain more. So it will widening and then increase your transfer of body heat to the environment. And then another one is insulation. So it will decrease your rate of insulation. So I already mentioned insulation is to reduce the flow of heat, right? So reduce the flow of heat. For in hot day condition, you need to increase the flow of heat, so your rate of insulation will be decreased. Okay. Another one, your sweat gland was starting to secrete more sweat, and then your evaporation of the sweat gland from skin surface will prevent the body cold temperature. So this is an evaporation cooling effect lah. And then your behavior response also will take bath, like you will bath in water moving to cooling area like that lah. And then in your cold day, what your happening is your heat gain center at the posterior hypothalamus. So this what is your heat load center at anterior hypothalamus. This what is your heat gain center in your posterior hypothalamus. Okay, so it will also send the nerve impulse into your effector. So one of the effector is your blood vessel. So it will starting to contract. So it will resulting in your vasoconstriction. Okay, vasoconstriction will reduce the blood flow and decrease the heat transfer. And also insulation will be increased, and then your hair evator muscle will contract to pull the hair up and straight. And then this will increase the thickness of the layer of air trap. Okay, not only that, thermogenesis also will be take place. So I think this is an explanation previously lah. And they also consists of bound fat between the shoulder, and then bound fat tissue will generate heat. And then not only that, the endocrine gland, the adrenal gland will also stimulate the adrenaline, and your thyroid gland will release the thyroxine, and your pituitary gland will also stimulate the thyroxine stimulating hormone, and also ACTH hormone to increase your metabolic rate. So the behavioral response also will take place So they normally, like, they will go to the warm place like that lah. Okay, so we already finished about the first topic. So now we go to the second topic. Okay, now we go to the second topic, which is our liver. Okay, liver. So I think everyone know what is liver. Okay, so firstly, liver is one of the organ, very important organ that play the role in detoxification, right, and also absorption process that you learn in SPM, right. So in SPM you will learn more detail about the liver structure. Okay, so normally, this one is a diagram that you see in the human anatomy structure. anatomy structure. So normally liver is composed. There are many liver lobules, and then each lobule will contain many liver cells like this. So normally in examination, you need to remember this kind of structure. So normally you will see that hepatocyte is the basic unit of the liver cell. So the basic unit of liver cell is known as hepatocyte. And then in liver, we know that we have three type of blood vessel, right? Portal hepatic artery, and also hepatic vein, right? So there are three type of blood vessel. So you need to know each function of blood vessel, and we also know that in liver we also have the bowel conduction, bowel ductus. Okay, so this is the explanation about the liver. So normally liver is the largest visible organ that locate on the right side of the body, immediately beneath of the under the diaphragm. So normally liver has a double blood supply to carry out its function, hepatic artery and also hepatic portal vein. So normally hepatic artery and hepatic portal vein, hepatic artery is to deliver oxygenated blood to the liver, and then hepatic portal vein is deliver nutrient blood from the small intestine to the liver. But hepatic vein is carry the oxygenated blood from liver to anaconda. So hepatic artery and hepatic vein are the blood vessel that normally occur in all type of organ. Artery is to deliver oxygenated blood. Artery vein, is carry the oxygenated blood. Just that in liver there are another special vein is to deliver nutrient blood from small intestine to liver. So this one is that you already learned in Stpm. Liver also play the function in food in the nutrient absorption, right? So normally it will deliver. So the absorption that done by the small intestine will some of the nutrient will be delivered through the hepatic portal vein to the liver. So we will see this kind of structure. Normally in examination they will give this kind of structure this kind of structure. So normally you will see this structure. The hepatic portal vein and the hepatic artery normally is connected to each other one. Normally your hepatic portal vein and your hepatic artery will be connected to each other one. Why? Because these two blood vessels are supplying blood into the liver. So they were in the similar location until that they were joined together. So the blood flow will flow from here, from left hand side to the right hand side. So normally this one, the another blood vessel will be occur at here. Hepatic portal vein will occur at here. So you refer to this diagram again. So you see that artery and your portal vein will be in the similar location, and then your vein will be at here. Then all these cell is we call as hepatocyte. All these cell that you see, all these we call as hepatocyte. And then and then you will see that the tube like this, the blood vessel inside between the hepatocyte is we call as sinusoid. And then on the hepatocyte there are the cell like this one we call as capsule cell. So you will see this diagram. So this diagram does not show but actually should be have. So normally the liver is divided into two main lobes, right lobe and left lobe. So each lobe contains the large amount of lobule. So each lobule contains many hepatocytes, And then hepatocyte contain nucleus, which in mitochondria, Golgi protein, and lysosome lah. Okay, and then the hepatocytes are tightly packed to each other, and their surface are contact with blood vessels that have microvilli, which are used to for material exchange. And then okay, the bunch of the hepatic vein that carry nutrient and arteries that carry oxygenated blood to the liver can be known as interlobular vessel. So normally so if you see this diagram, so the normally the bunch of hepatic vein that carry nutrient and the artery can be known as interlobular vessel. And then the blood is passed through the sinusoid, which drain the blood between the cords of hepatocyte. So along the sinusoid, material are exchanged between the blood and hepatocyte. So when they are the interchange between the hepatocyte, this one we call as sinusoid. And then the blood vessel that already joined together, we are called interlobular vessel. And then the interlobular vein or central vein is located at the center of the tubule to receive the blood from sinusoid and then pass through into the hepatic vein. So at here lah. So you see this diagram is more detailed. It will at the central vein, and then it will pass to the feed into the hepatic vein. Okay, and then the arrangement of the blood vessel, hepatic portal vein, hepatic artery, and the hepatocyte are from the functional unit we call as encynus. And then the hepatocyte also will produce bile and store in the gallbladder. And then the bile was secreted for hepatocyte transfer through the bile canaliculi between the adhesion layer of the separated and then to the bile duct. So if you see this diagram, so normally there are another layer, right? Another like the blood vessel, right? So this is another vessel, right? So this is not a blood vessel. This is a flow vessel that spatial only for bile to flow only. So normally hepatocyte will secrete the bile, and then they will flow through the vessel we call as bile canaliculus. So this bile will flow through this bile canaliculus and then go to the gallbladder and store at here. And then there are also the capsule cell found in the layer to form the reticular endothelial system. So normally it is as the elimination of the bacteria and all erythrocyte by phagocytosis. Okay, next we go to the liver function. So in our syllabus, we have two main liver functions that you need to know. Is about the carbohydrate metabolism and also the phagocytosis. Okay, next we go to the liver function. Okay, Okay, so in liver function we have two main of functions, which are carbohydrate metabolism and also protein metabolism. So under carbohydrate metabolism, you need to know about the glycogenolysis, and also gluconeogenesis. And protein metabolism, you need to know about the deamination, ornithine cycle, and also transamination. Okay, so actually this is one of the topic that you learn in semester 1, right? I think in semester 1 you learn about the carbohydrate metabolism, right? Krebs cycle, glycolysis, all that. So in semester 2, this one is only occur in our liver cell only. Other cell will not happen. Okay, so about the carbohydrate metabolism. Okay, I think everyone should be know of these two hormones, insulin and also glucagon. Okay, so normally insulin is play the role to decrease the blood glucose level to normal when your blood glucose level is very high, and your glucagon is to increase your blood glucose level when your blood glucose level is very low. Okay, so normally insulin is both are secreted from pancreas, but the cells are different. Insulin is secreted by the beta cell of islet of Langerhans in pancreas, but glucagon is secreted from alpha cell. Sorry. Yeah. Alpha cell. Sorry. This one, glucagon is alpha cell. So insulin is secreted from the beta cell, and glucagon is secreted from alpha cell of islet of Langerhans. So insulin, I already mentioned, is respond when your blood glucose is very high. So only glycogenesis is happening, and then glucogenesis is happening. And glucagon it was secreted when your blood glucose level is very low. You need to increase your blood glucose level to the normal. So glycogenolysis and gluconeogenesis will be happen. Okay. So this is the overall process, negative feedback mechanism that liver will be take place. Okay, now you proceed to the glycogenesis. So glycogenesis is a process to form glycogen. So glycogen, everyone know, right? Glycogen is one type of polysaccharide that store in the animal cell. So normally liver is play the role inside of this lah. So normally when the person have the high blood glucose level, your liver starting to proceed the glycogenesis with the help of the hormone insulin. So this is the structure, and then if you know that this is the more simple structure of the glycogen, so you know that glycogen is a highly branched structure, and then it contains 1 alpha 1 4 glycosidic bond and also 1 6 glycosidic bond to form the branched structure, right? So you need to learn about how the glucose molecule can be combined together to form this kind of glycogen. This can form like this one, this complex polysaccharide structure. Okay, so the first step of the glycogenesis is about to the okay, so the first step of glycogenesis is to you need to, your glucose molecule will be phosphorylated to become glucose 6-phosphate. So this one is similar to your first step of your glycolysis. You still remember, so you don't think that you already in sem 1, in sem 2 you need to forget all this. No, because in semester 2 you need to remember all this too. Glucose will be phosphorylated into glucose 6-phosphate using hexokinase enzyme. After that, yeah, but in glycolysis, glucose 6-phosphate will be isomerized to fructose, right? But in glycogenesis, glucose 6-phosphate will be isomerized into glucose 1-phosphate, meaning your phosphate group from the 6th carbon position go to the 1st carbon position. So this is isomerization process. Okay, next we go to step 3. So your glucose 1-phosphate later will react with another molecule we call as UTP. So the UTP full name is uridine triphosphate. So the UTP molecule will act as the energy activator to activate glucose 1-phosphate first before synthesize glycogen. So UTP is like the energy lah. It's just like the energy activator to activate glucose 1-phosphate. So it will bind toward the glucose 1-phosphate So later you will form this molecule. So you will form the uridine diphosphate glucose, UDPG. Okay, so normally glucose will be add with UDP to form UDP glucose by breaking up 2-phosphate group. So 2-phosphate group from the UDP will be break down, and then this enzyme that using is UDP glucose phosphorylase. Okay. Next, step 4 is the final step that the glycogenesis will be happen. Okay, so you already know that UDP glucose is already formed. Now you need to addition of UDPG to a going chain of glycogen is the next step in the glycogen synthesis by using glycogen synthase to form 1,4-glycoside. However, in this enzyme, the glycogen synthase cannot simply form a bond between two isolated glucose molecules. So when only have the simple one UDP glucose molecule, if you want to form 1,4-glycoside bond using this enzyme, it cannot directly form. So you need to have a primer. So one of the primer we are known as glycogenin. So this is a structure of glycogenin. So this glycogenin will act as the primer to initiate the glycogen synthesis first before the glycogen synthesis can be take place. So glucose residue will first link to the tyrosine hydroxyl, and glucose residue are successfully added to this first one. So normally glycogenin will be added to the UDP glucose and then link to this tyrosine hydroxyl group. Okay, after that they will have the activity here, and then UDP now will be released out. This molecule will be released out, and then another UDP glucose molecule will be add to this another UDP glucose molecule will be add to the carbon number 4 of the glucose to form 1,4-glycoside bond. So if you see this diagram, this is the initial structure that already activated from this glucose molecule. So you refer to this glucose molecule. This UDP later will be released out. Once it released out, it only will occur like this. So this glucose molecule already have a glucose molecule and with the glycogenin structure. And then since they are the primer at here, another UDP glucose molecule will be add to here, and then the number 4 of the carbon will be add with the number 1 of this new UDP glucose to form the 1,4-glycoside bond. So the glucose residue will link to this tyrosine hydroxyl, and the glucose residue are successfully added to this first one. Okay, so the glycogenin molecule itself acts as a catalyst for the addition glucose until they are about 8 of them linked together. Okay, so this process will be repeated 6 times again until they are the 8 glucose molecule is already linked together. So once there are 8 molecule already linked together, glycogen synthase will take over to form the 1,4-glycoside bond, and then your glycogenin will be detached. Okay. So finally, step is about the bunching step. So we know that glycogen is the highly bunched polysaccharide. Just now it's just about the formation of 1,4-glycoside bond. Now we need to form 1,6-glycoside bond. So normally this one is we have the enzyme called bunching enzyme. So if you see here, normally when the it will break down branching enzyme normally will break down the 1,4-glycosidic bond at the branch point and transfer this oligosaccharide fragment into another chain by catalyzing the formation of 1,6-glycosidic bond. So normally you see this diagram, you will see here they already this one already formed the branch, but when they form the branch at here, and then the branching enzyme will detect the branch point. They will detect the branch point. When they detect the branch point, so assume that this is the branch point. This is the branch point. They will break down the 1,4-glycosidic bond at here. So once they break down the 1,4-glycosidic bond at here, they will transfer this enzyme will transfer all these oligosaccharide fragment into the another change, and then this one maybe it will go to this change to form another branching change. So this one it will directly go to here, so it will form another branching change like this. So it will form the 1,6-glycosidic bond. So this is the final step of the process. Okay, so this is overall process of the glycogenesis lah. Okay, next is about the glycogenolysis. So just now is about the insulin, about the function of the insulin. When your blood glucose level is very high, it need to go through the glycogenesis to form glycogen to maintain your blood glucose level decrease into normal. Now when your blood glucose is very low, so you need to have a higher amount of blood glucose, right? So glycogenolysis is one of the pathway that break down of glycogen. So it will break down the glycogen to produce glucose molecule, right? So glycogenolysis is more simple compared to glycogenesis. Okay, so normally you have glycogen. So normally glycogen that link with 1,4-glycosidic bond can be cleavage by the glycogen phosphorylase through the phosphorylases to form glucose 1-phosphate. So it can directly glycogen, if you remember glycogen structure, it can directly for the 1,4-glycosidic bond, it can directly using the enzyme we call as glycogen phosphorylase. It can directly break down the 1,4-glycosidic bond and then to form glucose 1-phosphate. So it can directly break down like this. So it can directly form glucose 1-phosphate. So when they form the glucose 1-phosphate, it will undergo isomerization using the enzyme we call as phosphoglucomutase to form glucose 6-phosphate again. And then this glucose 6-phosphate will be undergo dephosphorylation to form back again glucose molecule. However, this one is directly, but when there are the branch structure like this, you see that there are the branch structure. You need to hydrolyze the 1,6-glycosidic bond. So how it is hydrolyzed? So normally they will have the transferase. So normally the glycogen phosphorylase, they will break down the 1,4-glycosidic bond for the branch structure. They will break down until they remaining the 4 residual glucose molecule at here. Okay, when there are the 4 residual molecule at here, we call as limit branch. The limit branch transferase enzyme will take place and then break down the 1,4-glycosidic bond at here. The last one, the 1,4-glycosidic bond of the last glucose, and then will transfer this like limit branch into the another change. Okay, so now it only left one residue at here, so that alpha 1,6-glucosidase will be take place what we call as debunching enzyme to break down the 1,6-glycosidic bond at here and then to form the glucose 1-phosphate. And then this glucose 1-phosphate will go to produce, and then this another glycogen change will be using the same enzyme glycogen phosphorylase to form glucose 1-phosphate. So this is the explanation. So transfer will break the unfold glycoside of the limit one, leave only one residue that link with 1,6 glucoside bond. Okay, so this is overall process of glycogenolysis. So this is one of the process. This is the initial process of the glucagon hormone will be initiated when your blood glucose level is very low. You need to break down the glycogen. However, when the people is in very fasting state, your glycogen is already used up. Or there are another metabolism we call COVID cycle. Later we will learn it. When your glycogen is totally used up, you will proceed to gluconeogenesis. Gluconeogenesis is the formation of glucose from pyruvate. So it is all. Another definition is that formation of glucose from non-carbohydrate resources. Any non-carbohydrate resources like protein molecule, acid, lipid molecule, amino acid, glycogen, all these is considered as non-carbohydrate resources that will form glucose molecule. This process we call as gluconeogenesis. So this is the overall process of gluconeogenesis. So actually, if you see here this diagram, I think everyone is very familiar. So we can say that gluconeogenesis in overall is the reversible reaction of glycolysis. However, there are some minor change. If you still remember your glycolysis reaction, that in first step, third step, and the final step of the glycolysis is irreversible reaction. It only can go through one reaction only. So so because of this gluconeogenesis need to take another method to complete it. So how it is complete? So if you see here, your final step, your first step, and your third step, this is the process. So normally, if you see here this diagram, so normally it is a reversible reaction for the glycolysis in general. So normally, if you see here, lactate, amino acid, and your glycerol will go toward this. Will be as the intermediate. Go toward this. Go toward the process of gluconeogenesis, and then directly to form glucose molecule. Okay. So what are their difference is during the first step of the gluconeogenesis, you see here, this is the carboxylation process, meaning that your pyruvate will not directly to form the phosphoenolpyruvate, same as the glycolysis, because it is an irreversible reaction. So you need to form the oxaloic acetate first, and then this process is occur in the mitochondrial matrix. It is not occur in the cytoplasm. You need to remember, you know that glycolysis is occur at the cytoplasm, right? But the first step of the gluconeogenesis is occur in the mitochondrial matrix, meaning that your pyruvate need to enter into the mitochondrial matrix first. So normally your pyruvate will undergo carboxylation to form oxaloic acetate using the pyruvate carboxylase, and then this process requires ATP. Next step. Okay, so after you already form oxaloic acetate, you were using the enzyme. PEP, KP carboxykinase, using GTP to form phosphoenolpyruvate or phosphoenolpyruvate. So this one you will release carbon dioxide molecule. So this one is what we call as decarboxylation. First step is carboxylation process, and then you will go through decarboxylation process to form phosphoenolpyruvate. Okay, after that After you form a strong endopyruvate, you will give the similar reaction like glycolysis. You will form 3-phosphoglycerate, 2-phosphoglycerate, 3-phosphoglycerate. You will reversible reaction using the same enzyme that you learned in semester 1. If you already forget, please recap it. I think your semester 1 book is not you not throw yet. You need to remember. If the examination want you to about this, you need to mention all this. Okay, 3-phosphoglycerate, and then you go to the 1,3,5-phosphoglycerate, you go to glycerate 3-phosphate, and then dihydroacetophosphate, until you go to the fructose 1,6-biphosphate. Okay, so you go to fructose 1,6-biphosphate, you need to form fructose 6-phosphate. So this is the dephosphorylation process. You are using this enzyme lah. After that, fructose 6-phosphate, you will form glucose 6-phosphate. Glucose 6-phosphate, you need to form glucose molecule. So glucose 6-phosphate, you will form glucose molecule using glucose 6-phosphatase, form glucose molecule. So this is the overall process of gluconeogenesis. Okay, that is about the COVID cycle. So normally COVID cycle is doing when you are exercising, your glucose molecule under glycolysis to form pyruvate, right? This is the anaerobic respiration that you already learned in semester 1, right? That will form lactic acid, and then finally you will form lactic acid. Okay, this lactic acid later will be transferred from muscle tissue into the blood vessel, and then transfer into the liver, and then this lactic acid will undergo reversible reaction to form pyruvate, and then it will undergo gluconeogenesis to form glucose molecule, and then in the overall process of glucose molecule, you will require 2 ATP and also 2 GTP in overall process of the COVID cycle lah. Yeah, so this is the overall process of COVID cycle you need to know. And then this glucose molecule will be transferred back into the muscle tissue, and then this process will be repeat again until your lactic acid is disappear lah. You're not doing the exercising again. If you are doing exercising again, your glucose need to produce lactic acid again. This lactic acid will produce back glucose again, so it will repeat again lah. Okay, now we go to the protein metabolism. Okay, now we go to the protein metabolism. So normally protein metabolism involve about the first step is involve about the deamination process. Okay, so deamination is the removal of amino group from amino acid to form ammonia, which is very toxic and soluble. So normally if you see this one of the example, so normally only one of the example that are using glutamate. So normally glutamate will be deaminated to remove the amino group from the amino acid to form the ammonia or ammonium. So when the ammonia dissolve in water, it will form ammonium ion. So this is the process. Ammonium ion is very toxic and soluble structure that we don't want. Our human cannot store ammonia one. We need to excrete out the ammonia. Okay, so after deamination, later we will go to the ornithine cycle. So so the first step is about the first step of the ornithine cycle is condensation reaction. where you already form the ammonium ion. You already form ammonium ion, right? Okay, for ammonium ion, and then this ammonium ion will react with carbon dioxide, and then water molecule is added. So that is why that it is condensation reaction, because it requires water molecule, and then it will use 2 ATP to form carbonyl phosphate, and then this process is occur in the mitochondria. So the first step and the second step is occur inside the mitochondria. Now mitochondria matrix. Okay. After that, when the carbonyl phosphate is formed, it will transfer this carbonyl group to ornithine. So we will see this structure. Ornithine is the structure like this, and then this carbonyl group will be transferred into the ornithine to form citrulline. So you will see, refer to this the C under this C and H, under belong the NH is the structure of ornithine, and then this C and H2, all this is come from the carbonyl phosphate. So it will release one phosphate group. The phosphate group will be released. Okay, next is citrulline. Citrulline will be formed aviginous-succinate. So meaning that citrulline will react with the aspartate, one of the amino acid. It will react the aspartate to form avininous-succinate, and then this process will require 1 ATP molecule. And then starting this process will occur in the cytoplasm of the liver cell. So meaning that your citrulline will be transferred from the mitochondria into the cytoplasm. So you need to mention first, you need to mention that the citrulline will diffuse into the cytoplasm, and then once it reaches the cytoplasm, citrulline will react with the aspartate to form argininosuccinate. Okay. After aviginous-succinate, it will undergo the dissociation process. It will dissociate fumarate, and then to form arginine. So you will see that the fumarate structure, this structure, you see this one, because you will remember that aviginous-succinate, this one is come from the aspartic acid, and then this one come from aspartic acid, and then this one will be rearranged again to form fumarate, dissociate to form fumarate. Okay, so this fumarate, and later it will enter into the Krebs cycle lah. But then others will form arginine. Okay. After that, arginine, what will happen to arginine is that arginine will undergo hydrolysis reaction. It will hydrolyze to form urea. So this is the urea that we don't want. That will be excreted out through kidney. The toxic that we don't want, because it contains ammonia, right? You see here, contain ammonia group. Ammonia group, I already say it is very toxic, that cannot be stored in the human. Right? Our human don't want this. We need to excrete it out ammonia. So it will excrete it out in the form of urea. So your urea will be arginine will be hydrolyzed to form urea, and then to form back again ornithine. and then this cycle is repeat and repeat again. It will repeat the cycle on it. It will repeat again, react with the carbonyl phosphate again to form citrulline, and then this cycle will repeat and repeat again. Okay, so this is about the ornithine cycle lah. And then the last one of the reaction in protein metabolism is about the transamination. So normally transamination is about the protein synthesis of new amino acid by the enzymatic transfer of amino group between the amino acid and the keto acid. So normally this is the general formula. Normally you will begin with the glutamic acid. Glutamic acid is the key component, and then you will react with keto acid to form the alpha keto acid, and then form another one is another alpha amino acid. Okay, next we go to the osmoregulation in animal. Okay, now we need to know about the mammalian kidney lah. Okay, so this is the overall structure of mammalian kidney. This is the human anatomy, right? So you know that our kidney is like a bean shape, right? And then this is our kidney, and then we know that in our kidney we have two areas, right? Either is medulla area, the upper region is our medulla area, and then the upper region is our cortex, vena cortex, and then our lower region is we know as vena medulla. And then we know that the nephron is the basic unit of the kidney, right? Nephron. And then we know that our kidney contain Bosman capsule, glomerulus, presomal tubule, loop or handle, distal tubule, and also collecting duct. Okay, so this is the overall structure of mammalian kidney. So I don't need to explain more lah. So you can read yourself. Okay, the most important part is about this urine formation. Okay, so normally in urine formation we have three main process, which are ultrafiltration that occur in glomerulus, reabsorption occur in the tubule and lobe of Hindle, and secretion that occur in collecting duct. Okay, so this is the overall structure of nephron that the urine formation will be conducted. So you know that in the first process, the arteriole, the blood capillary, your blood structure, your blood vessel that is about the arteriole will enter the into the Bosman capsule, right? Enter into the glomerulus, and then it will undergo ultrafiltration to filter out some of the component, and mainly is about the nutrients, salt, water molecule, all that will be filtered out, and your waste product like urea, uric acid, all these will be filtered out into the Bosman capsule. After that, Bosman capsule will go to the through the ultrafiltration. So ultrafiltration is occur at here. After that, all the fluid, all the glomerular filtrate, the filtrate substance will enter presomal tubule. So when it enter the presomal tubule, reabsorption will be occur. So throughout the presomal tubule, throughout the loop of Hindle, and also distal tubule, reabsorption will be occur at here. And then after that, finally it will enter the collecting duct. Okay, so collecting duct is a process of the secretion. See, this is the final process. It will excrete out all the waste product, and then finally it will form urine. Okay, first is about the ultrafiltration. So force the small molecule out of the blood plasma at the glomerulus capillaries and enter into the Bosman capsule. So you see this structure. This is the afferent arteriole. So normally the blood will flow from the afferent arteriole. So the blood from the afferent arteriole has a high hydrostatic pressure. So when it enter into the Boltzmann capsule, ultrafiltration will be occur to forcing out all the components like water, mineral, salt, and your waste product, and also glucose molecule, protein, amino acid. All of them will be filtered out from the glomerulus capillary into the Boltzmann capsule. After that, it will enter the proximal tubule. Okay, so we will learn more detail about the glomerulus structure. Okay, so we know that glomerulus capillary are the network of capillary from afferent arteriole and rejoining from more thinner afferent arteriole. So normally the capillary are made up of many layers of the endothelial cell, which contain many pores that allow the passage of all blood components. And then the membrane, the endothelial membrane, is covered by the cell we call as podocyte. Covered by the glomerular basement membrane, they make up of protein, and then it separates the glomerulus from the capsule, and then it also prevents the blood cell, platelet, and blood plasma protein enter the Boltzmann capsule. Okay, so you see this. Okay, so if you see this diagram, so you see that there is a capillary lumen, and then there is the layer of the endothelial cell layer, and then this is the podocyte layer. So normally ultrafiltration will be happening because one of the reasons is high hydrostatic pressure, and then another one is large amount of glomerular filtrate is large surface area for filtration provided by the highly coiled glomerular capillary. So you will alert that our glomerular capillary is highly coiled to provide the large surface area, and then also great permeability of the glomerulus capillary that contain many small spores. Okay, so normally what component will be filtered? So normally the wall of Boltzmann capsule or contact with the capillary consists of specialized cell called podocyte. So podocyte play the role in the ultrafiltration. Why? Because this cell will have the enormous cytoplasmic extension called foot process that cover on the glomerular basement membrane. So normally this foot process will be separated by the narrow gap called filtration slit. So you see this diagram, there are many filtration slit, right? So this is the process that the place that all the component can be filtered out. So if you see this one is more detailed. You will see this purple color we call as podocyte, and then there are the many filtration slit at here. Okay, so normally this porous wall can wall the glomerular capillary and also filtration slit can allow the small solute to dissolve. So normally the glucose molecule, amino acid, salt, sodium salt, potassium, bicarbonate, and your urea acid, all this will be filtered out. Okay, and then this one is more detailed. You will see that endothelial cell, if we know that endothelial cell is already contain many spores, already can make the diffusion occur, and then basement membrane, and then you will see that in the podocyte there are also the filtration slit at here, so it can allow the many components to be filtered out. Okay, next is about the V adsorption. So normally V adsorption is occur along the tubule, loop or hinder distal tubule, and even though in collecting duct you also have V adsorption. So normally it is a process of filtrate adsorption after the filtration process occur. Okay, so now we go to one by one. It's about the V adsorption in proximal tubule. So in the proximal tubule, normally all of the components like glucose molecule, your amino acid, chloride ion will be at V adsorbed. So that is why why that in your urine you will not contain any glucose molecule, any amino acid lah. If you have glucose molecule, amino acid, then you should have something probably happen in your kidney lah. So mainly is because of plasma tubule. Okay, so mainly or because of convoluted plasma tubule. So normally at the plasma tubule, the tubule is lined with a single layer of epithelial cell with extensively microvilli on the plasma membrane. So you see that your plasma tubule is contain is like the finger-like projection. They contain the many microvilli, and then at this time all your glucose molecule, your sodium ion, your amino acid will be diffused out from the plasma tubule into the epithelial cell first, in the vena epithelial cell. So normally the outer membrane of epithelial cell are covered with basement membrane. So they are the epithelial cell. We know that epithelial tissue that you learn in semester 1, kidney, the plasma tubule is containing the epithelial cell, right? Okay. So normally salt like NaCl will be filtered out into the epithelial cell by facilitated diffusion, and then will be transferred to the interstitial fluid by active transport. So for the sodium ion and the chloride ion, for the salt like sodium ion will be facilitated diffused into the epithelial cell first, but later it will using active transport, using sodium potassium pump to pump the sodium ion out from the cell. Okay, and then this will transfer a positive charge out of the tubule, drive the passive transport of the chloride ion. Okay, and also not only salt, your water will also diffuse out from the cell, and then your glucose, amino acid, potassium ion will diffuse out from the cell, either is passively transport or actively transport lah. And then the waste product like urea will diffuse into the plasma convoluted from interstitial fluid during non-selectively filtration. Okay, and then the other product will remain in the filtrate. So in conclusion, your sodium ion will be diffused out, and then diffused out into the epithelial, and then it will diffuse out again using the active transport sodium potassium pump. And then another one, glucose molecule, amino acid, chloride ion also will be diffused out again, O2, and then your water molecule also will be diffused out. Okay. Another one is about the reabsorption in proximal tubule. So this is a continuous process. So normally the reabsorption down the concentration gradient, and then will initiate reabsorption of urea by diffusion. The small protein in filtrate are reabsorbed by the pinocytic vesicle, and then the primary adsorption within the cell attached to this vesicle, and then this hydrolytic enzyme will digest this protein into the amino acid lah. Okay. And then about the reabsorption in the loop of Handel. So in loop of Handel we have two pathways, either is descending or ascending. So in general, in descending pathway, only water is absorbed, only ether is be absorbed. But in ascending pathway, there are two segments, either is in thin segment or thick segment. In thin segment, your salt and HCl will be reabsorbed by using facilitated diffusion, passively transport. But in thick fragment, it is actively transport. Okay, so why it is happening like that? So normally in descending pathway is very permeable to water lah. So no channel for salt, no channel, so sodium chloride cannot be absorbed again. So normally the interstitial fluid is hyperosmotic to the filtrate. So your interstitial fluid at here is very hypertonic to the filtrate one, so that your water molecule can be filtered out. Okay, so that the condition is maintained along the descendant lead because the osmolarity of the interstitial fluid increase from the cortex to medulla. So you will see this diagram. You will see that the color is more darker, right? So at this point your solute potential is very high. So along this point, your water molecule will be absorbed, will be reabsorbed very. Your rate of osmolarity will be very high along when you reach the more deeper of the loop or handle. Okay, now we go to ascending. So ascending, I already mentioned there are thin segment and also thick segment, right? So normally in thin segment, it is highly concentrated of filtrate. Filtrate. So normally because you already at here, your water molecule, most of the water molecule is already reabsorbed. So now at this part, your filtrate is highly concentrated, so that your salt in the filtrate can be directly reabsorbed from the loop or handle into the interstitial space by using the facilitated diffusion. Okay, so this movement of NaCl can help to maintain the high osmolarity of the interstitial fluid, because I already mentioned, because in during the descending pathway, you need to maintain your interstitial fluid is hyper osmolarity, because your water molecule need to diffuse out. So if you want to maintain hyper osmolarity, then salt you need to diffuse out again. So when the salt is diffused out again from the loop or hinder, then it will maintain the hyper osmolarity in the interstitial space lah. So this is like the cycling process lah. So normally in thick segment, so normally sodium chloride is filtered out, but now is using active transport. Why? Because we already know that at here most of the sodium chloride is already absorbed. But for now, if you still want to make the NaCl to be reabsorbed again into the interstitial space, now the interstitial space is hyperosmolarity, hypertonic condition. So in simple diffusion, your salt cannot diffuse out in using facilitated diffusion. So you need to use active transport. So normally you will use the active transport to maintain the high concentration of the interval of the kidney, and enable kidney to form concentrated urine. Also, such counter current system expend energy to create the constant gradient called counter current multiplier system. Also, the filtrate is actually hyperosmotic to body fluid by the time it reaches the distal tubule. So at this time, mostly your salt will be reabsorbed. So now your filtrate was starting to become hyperosmolarity. So as the descending pathway, although many water is reabsorbed again, but sometimes if you drink many water, your water not all water will be reabsorbed. Some of the water still remaining in the filtrate. Okay, in the filtrate. Okay, so now we go to distal tubule. So normally in distal tubule, it is a key role, play the role in regulating potassium ion and sodium chloride concentration. So normally your potassium ion was secreted into the filtrate, while your sodium chloride will be absorbed from the filtrate. So normally this one is about the sodium potassium pump. The sodium potassium pump will be carried out, meaning that sodium ion will be actively transported out, and then your potassium ion will be actively transported out into the filtrate. So the filtrate now is hyperosmotic. So so the distal tubule also contribute the pH regulation. Later we will see it. Okay. So next, we go to the collecting duct. So collecting duct not only play the function in secretion, it also play the function in reabsorption. Okay, okay, okay. So normally in collecting that, more of the salt, your salt will be reabsorbed again, from the more salt and HCl will be reabsorbed again from the filtrate, and also your your water molecule will also be reabsorbed. And then finally we go to secretion. So is the expulsion of unwanted substance lah, and then your excess ion. So normally are actively secreted into the filtrate. So normally that your jar, your alcohol and your urea, all this will be eliminated lah. Okay. So normally now I go to the ADH hormone that play the role in osmotic regulation. So this is the summary process. So normally ADH is secreted from the posterior pituitary gland, and then it will accept the signal from the hypothalamus, which the osmotic receptor is located. So normally osmotic receptor is stimulated because of blood osmolarity increase. So normally meaning that when what are meaning the blood osmolarity, meaning that your blood osmolarity is the like the your blood content, your blood pressure and your blood water content. So normally when your blood pressure is very high, or your water content is very low inside in your blood, it will osmotic receptor will be stimulated, and then it will send the impulse to your posterior pituitary gland, and then posterior pituitary gland will release the hormone ADH ADH, and then this ADH hormone will increase the permeable of your collecting duct, and also this the tubule to reabsorb more water molecule, to reduce your blood osmolarity to become normal lah. Okay, so this is the overall process. Okay, so normally when your blood osmolarity is high, so I already say your osmotic receptor is detected, and then it will secrete ADH and transport to the posterior pituitary gland, and then it will release ADH. So ADH will transported into by bloodstream into the kidney tubule, and then it will increase the permeability of water molecule on the surface of distal convoluted tubule and collecting duct. And then aquaporin water channel will fuse to the plasma membrane of the kidney, so it will increase the water reabsorption. So water will flow from the filtrate into interstitial fluid, and then directly go to the blood capillary. So this will cause the urine volume decrease and produce highly concentrated lah. Okay. And then this one is about the response of ADH to epithelial cell. So normally this one is similar like the mechanism, one of the example of mechanism in non-steroid hormone lah. Because ADH is one of the mechanism of non-steroid hormone, right? So it is the second messenger activation mechanism that you learn in chapter 9. Okay, so I think the initial process I don't need to repeat lah. I need to focus on the protein kinase. So once the protein kinase already activated, it will induce the cellular response, meaning that it will produce the activate phosphorylate, cause the vesicle with aquaporin to fuse with plasma membrane of epithelial cell. So it will generate more storage vesicle that contain aquaporin water channel. So this aquaporin water channel is actually make the collecting duct or your distal convoluted tubule more permeable to water. So when there are many water channel at here, storage vesicle, so they will go through the fused with the plasma membrane of epithelial cell. Now aquaporin channel will enhance the reabsorption of water from collecting duct to interstitial fluid and then to blood capillary. So it will produce many aquaporin channel, so that the water molecule not only will be absorbed, will be diffused through the plasma membrane, it can directly diffuse through the aquaporin channel, so it can facilitate the diffusion. Okay, so normally when the osmolality is low, your ADH hormone will not be stimulated or less stimulated. So your collecting duct and your distal tubule will not permeable to the water molecule, so the normality your urine volume will be very dilute Okay. And then another one is about the aldosterone. So not only IDH, another hormone is we call aldosterone also play the role in osmoregulation. So normally aldosterone also is more focused on blood pressure. So if you see here, so normally aldosterone is secreted by adrenal gland. So normally aldosterone will make more sodium ion, and your more water molecule will be absorbed at the distal tubule to increase the blood volume, so that your blood volume and your blood pressure will be remaining normal. Okay. And then this is our overall process. Later we will see it. Okay. So normally why aldosterone is very important. So normally ADH is only respond to change in blood osmolarity only, but it cannot respond to salt and body fluid are lost at the same time. So meaning that when your fluid is lack of salt, or your body fluid is low, is low, it cannot detect it. ADH cannot detect. So you need aldosterone. So you need to the endocrine system we call as renin-angiotensin aldosterone system, RAAS, to regulate this problem. So normally RAAS is respond to decrease of blood volume or your pressure and increase of blood osmolarity with ADH. So actually is cooperate with the ADH hormone. Okay, so normally if you see here, RAAS involve JGA, that is specialized tissue around the afferent arteriole. Okay, when your blood volume or your blood pressure is decreased, your JGA will starting to release enzyme renin. And then you will see here this diagram. So normally if your blood volume your blood volume or your salt is deficiency, you will cause the blood volume decrease and also cause the blood pressure decrease, so that your juxtaglomerular cell, this JGA in the kidney was starting to secrete renin enzyme. Okay, renin enzyme. And then at the same time, your liver will also secrete the enzyme called angiotensinogen. And so it will angiotensinogen. So renin will catalyze angiotensinogen to form angiotensin 1. So this is a plasma protein. Renin enzyme will catalyze the angiotensinogen to form angiotensin 1. So this angiotensin 1 will cause the lung to secrete angiotensin converting enzyme we call as ACE. And then this ACE enzyme will increase the angiotensin 2 secretion. So and ACE will catalyze angiotensin 1 to angiotensin 2. ACE enzyme will catalyze angiotensin 1 to form angiotensin 2. So this angiotensin 2 will play the role in these three roles. Either it can play the role about the vasoconstriction of arteriole. So when there are the vasoconstriction, the blood vessel will be contract, so your blood pressure will increase until it return normal. And another one is it will stimulate the adrenal gland to secrete aldosterone. Okay, so when your aldosterone is stimulated, it will increase your sodium ion and water reabsorption. Adsorption increase the secretion of potassium ion and hydrogen ion into urine. So it will increase the adsorption of sodium ion and also water adsorption, and then it will increase the blood volume. And when your blood volume increase, you will increase your blood pressure. Okay, so this is the overall process. So the most process is this. The initial process is this JGA cell was starting to secrete renin enzyme, and then at the same time your liver also will secrete the protein we call as angiotensinogen, and then this enzyme will catalyze angiotensinogen to produce angiotensin 1, and then this angiotensin 1 will stimulate the lung to secrete an ACE enzyme. So this ACE enzyme will catalyze angiotensin 1 to form angiotensin 2. So angiotensin 2 will have two pathways. Either it will contract the blood vessel to increase the blood pressure, and it will stimulate also the adrenal gland to stimulate the secretion of aldosterone, so that it will increase the sodium ion and water reabsorption. Okay, now we go to the pH regulation in tissue fluid. So normally this one is similar happening what you happen in the chapter 7. If you still remember your chapter 7 gets a change carbon dioxide transport in form of bicarbonate fluid. So how it is regulate. So normally in this is the example I proceed this in distal convoluted lumen. So normally when your pH concentration is very high, you will cause the pressure of hydrogen ion, right? That you learn in chapter 7. Okay, so when your carbon dioxide is very high, it will form the carbonic acid, and then it will form the hydrogen ion. So now your pH value is very low. So this hydrogen ion later will be diffused out from the cell, from the active tissue into the lumen, and then this hydrogen ion will react with the HPO42 negative to form H2PO4 negative, and then this H2PO4 negative will be excreted out. And then hydrogen ion will form HPO42 negative to form H2PO4, will act as a buffer to hydrogen ion, and then this will be secreted out into the collecting duct and excreted as urine. And that if the blood is too acidic, the hydrogen ion was secreted out by a transport from blood into tubule. Besides that, the epithelial cells are stimulated by synthesis ammonia. Ammonia will act as a buffer to trap the hydrogen ion to form ammonium ion 2. So you will see this ammonia ion. Okay, so this is about the pH regulation. Another last one is about the osmotic regulation in plant. So normally first one is about the importance of transpiration. So normally transpiration you already learned in chapter transport, right? So I think I don't need to mention about lah. So normally transpiration is very important because it helps to draw water and mineral ion, mineral salt and water absorbed by root cell by transpiration pool, transport water soluble organic substance, provide a cooling effect, play a major role in water cycle, water loss, high water potential, and then support photosynthesis, maintain the gas exchange, all this lah. Okay, and then we know that in plants, some of the plants they have xerophyte. So xerophyte are the plant that can adapt to survive at the hot, dry climate life area where the water supply is very short to supply. So example of the plant is cactus lah. Okay, so normally the stomata of the xerophyte is differ from the normal plant. So normally is little amount, the stomata number is very little, and then normally this reduce the surface area of leaf, resulting low rate of transpiration, and normally the stomata is sunken shape, and this make the stomata can trap the moist air, and then stomata is closed during day and open during night. Okay. And about the morphological adaptation of xerophyte, so normally the roots of these plants is very long, and then the stems are very flat in structure and very small and very thin, and then the plant surface are covered by tiny hair to reduce airflow, and then the leaf of the plant are very succulent leaf which is very thick that contain thick basic cuticle. So normally is to reflect to sunlight and reduce evaporation lah. Okay. About the physiological adaptation, normally the root cell have low water potential and high osmotic concentration lah, so that it can absorb water made frequently, and then the acetic acid is produced to close the stomata, and then the root can store many nutrients, and then the rate of growth is very fast when there is water during that time, and then the root stem leaf are store large amount of water. And another plant is we call as halophyte. So this plant is normally example is mangrove plant that normally can be survived at high salt concentration area, normally is near the sea lah. Okay. And then the morphological adaptation of this plant is they have the thick layer of cuticle to reduce rate of transpiration, also contain succulent leaf to store large amount of water, and then stomata is very little in amount, and then also have tiny leaf, tiny hair, and the spatial structure is it contains ionatophore for gas exchange, and then the leaf had the toy to remove excess salt, and then also have sunken stomata, and then the bulb root and the buttress root for anchorage and support for the moldy soil. And then for the physiological, normally it carry out viviparity to increase the chance of survival, and also accumulate salt actively into the plant to able water can be drawn into the cell by osmosis, and then secrete acid salts by the toy, and also can produce acetic acid lah. Okay, so that's all for this chapter. So now it's already 9:48. Okay, so we will take a break for 30 minutes. We will continue the next chapter immunity, chapter 12, on 10:30. Okay, so see you later. So have a break. testing. Okay. Okay. Okay, so now it's already 10:31. Okay, so we will start our next chapter, chapter 12, immunity. Okay. Okay, so for this chapter is regarding about the immunity. So there are four topics, no mistaken. You will learn about immune system, and then development of immunity, and then about concept self and non-self, and final one is about immune disorder lah. Okay. Okay, so for first one is about immune system. So we need to recognize our immune system. Okay. Okay, now we go to basic overview of our immunity. So normally immunity is defined as the resistance to disease, specifically to the infection disease. So it is the not process it is the terms that we define as it is the we can say it is a response of our body that resistant to our disease. So normally in immunity, as we know that you learn in Stpm, we know that we have first line defense, second line defense, and also third line defense, right? And then for first line defense and second line defense, we are known as innate immune response. So normally it is as the mediate the initial protection against infection lah. But in our Stpm syllabus, we will more focus on third line defense, which is develop more slowly and mediate later, even more effectively, highly specific in defense against infection. So the third line defense is the adaptive immune response. This is what we will focus for first line, second line, because you already learned in Stpm. So yeah, so you don't need to focus lah. So in our syllabus, we only focus about adaptive immune response only. Okay, now we will need to go to the lymphatic system. So as we learn in chapter 8 transport, you know that lymphatic system is important in also transport, right? One of the function is in the transport, that it can formation of interstitial fluid and then regulate the lymph again into the blood circulatory system. Okay. Okay, so lymphatic system actually is also play the role in the immune response too, especially in adaptive immunity response. So normally in lymphatic system, there are many lymphoid organs. If you can see this diagram, like bone marrow, thymus, spleen, lymph node, tonsil, all these are the main lymphoid organ that play the role in immune response, especially in adaptive immunity. Okay. So normally one of the organ is we call as bone marrow. So normally bone marrow is one type of primary lymphoid organ that supports cell renewal and differentiation of blood of the hemostatic stem cell, which is the stem cell of the blood cell. So normally bone marrow, we know that bone marrow is the site production of the blood cell, right? So normally bone marrow play the role in adaptive immune response. So normally all the blood cell is produced from bone marrow, but in the main role that we need to know is about lymphocyte, because lymphocyte is play the role in adaptive immune response that we will learn later. So normally all the cell will be produced in the bone, all the blood cell will be produced in bone marrow, but the maturation are differ, meaning that only B cell will be matured in bone marrow, but T cell, although it is produced in bone marrow, but later it was transported into the thymus for maturation. So T cell will be matured in thymus. So what is B cell and T cell? Like this one, I assume you already know, because you are Ulangan. I assume you already know that, so I don't need to say more what are B cell and what T cell. Okay, and then there are also 4 cells occur in the bone marrow: osteoblast. I think everyone should know. It is the bone cell, right? The basic unit of the bone cell that generate the bone and also control the differentiation of HSC. And then there are also the endothelial cell. Endothelial cell, everyone is know, it is the one of the surface layer of the blood vessel that also regulate HSC differentiation. And then reticular cell is the cell processing connecting cell to the bone and blood vessel. And another one is sympathetic neuron that release control and release of hematopoietic cell from the bone marrow. So this is the structure of the bone marrow. So you can see that there is reticular cell, there are HSC, ISC, osteoblast, and also sympathetic neuron. Okay, another main organ or lymphoid organ is we call as thymus. So normally thymus is also primary lymphoid organ that located in the thoracic cavity above the heart only. Above the heart, they play the major role in maturation of T lymphocyte. So this is the place that T lymphocyte develop. Major T lymphocyte is produced in bone marrow. After that, it was transported into the thymus for maturation. And then these are the process of the T cell development. Okay, T cell precursor, which still retain the ability to give rise the multiple hematopoietic cell type, travel by the blood from the bone marrow to the thymus lah. So after T cell that produced in the bone marrow will be transported into the thymus, and then the thymus have the environment, special environment can make the immature T cell to generate unique antigen receptor known as T cell receptor. You will generate the T cell receptor on the surface of T cell, and then selected on the basis of the reactivity to self-emancipate peptide complete expressed on the surface of thymoid soma cells. And then this tomocyte will bind to self-emancipate peptide with an intermediate affinity, undergo positive selection. So this is the overall process of T cell development. So this one later I will be explain more detail lah. So this is I just want to mention about the function of the thymus only. This is just the overall only. Okay, and then another one, an important organ is lymph node. So lymph node is considered as secondary lymphoid organ lah. So normally is located in the on the whole part of the body. Any part of the body in the lymphatic system will contain lymph node. So normally lymph node is packed with lymphocyte, macrophage, and dendritic cell. So the major role, this is the major role that play major places to play the adaptive immune response to activate B cell and T cell. And then lymph node also will provide the environment, the encounter between the antigen and lymphocyte, and productive organized cellular and humoral immune response. And then if you see the lymph node contain the cortex, contain the cortex and also parallel cortex. So the blue color one, you see the blue color is the cortex, and then the yellow color one at the middle one is parallel cortex, and then the purple color one is medulla. So normally cortex is a place it containing B cell, macrophage and dendritic cells. But the parallel cortex, the yellow color one, you are containing T cells. And then the medulla is the innermost layer where the lymphocyte and the lymphocyte acid the lymph node. These are places that the lymphocyte will be acid lah. Okay. And then antigen normally, because I already mentioned, lymph node is play the role in adaptive immune response. So normally antigen will be transferred from the infected tissue to the cortex of the lymph node by the afferent dendritic vessel. So there are the afferent dendritic vessel at here. And then antigen will enter either in particular form or it already processed in the antigen presenting cell lah. Okay. After that, particular antigen can be trapped by resident antigen presenting cell in the subcapsular synapse or the cortex, and it can be passed through to other antigen presenting cells including B lymphocyte. So normally this antigen, if the antigen is a particular antigen, it can be trapped in the cortex, and then it can be passed to other antigen presenting cell. So normally there are also antigen presenting cell to trigger the activation of the T lymphocyte. Alternatively, particular antigen can be processed and presented as the peptide MHC complex. Peptide MHC complex on the cell surface of resident dendritic cells. Okay. And another organ is spleen. So normally spleen is situated in the high left hand side of the abdominal cavity, and it is a large ovoid secondary lipid organ. So normally it plays major role in mounting immune response to antigen in bloodstream. So the role in compared to the lymph node is differ. Lymph node is to regulating the adaptive immune response to activate B cell and T cell, but for the spleen is to mounting immune response to antigen. Okay. So compared to, so what are the difference between the lymph node? So lymph node are specialized to encounter between lymphocyte and antigen drained from the local tissue, but spleen is filter the blood and trap the blood-borne antigen. So meaning that lymph node is already is the placing to already can do the immune response already. The lymphocyte is already can starting to do the immune response, but in spleen it is specialized in filter the blood first. It make a filtration first. Filter the which kind of blood is containing antigen, which kind of blood is not containing antigen. So if they are containing antigen, they will trap it. If not, they will pass through lah. So it like the filtration lah. So normally spleen is particularly in response to systemic infection. So normally the blood that containing antigen will be carried into the spleen through the splenic artery and out from the splenic vein. So if you can see this diagram, so normally the blood, they will pass through at here, and then pass through at here. And then it will go through out from here. So they will have a filtration process at this. So normally you will see here spleen red pump is a network that network of sinusoid populated by red blood cell, macrophage and cell lymphocyte. So normally it is a site where the old and defective red blood cells are destroyed and removed. So we will see here, this is the red part. Okay. So normally there are also white part. You will see here, this is the white part. White part is the space at the middle part one. marginal place, and the PALS, the blue color and the brown color places, is what we call as white part. Okay, so white part is the surrounded bunch of the splenic artery, consists of the PALS populated by T lymphocyte as well as B cell follicle. So normally this zone is to specialize the macrophage and B cell, which are the first line defense against the blood-borne pathogen. And then another organ is the mucosa associated lymphoid tissue. So there are 3 organs here: tonsil, appendix, and Peyer's patches. Peyer's patches. So normally this is the it is actually a group of different lymphoid tissue that group together that defends vulnerable mucosal membrane surface as a major site of the entry of most pathogen. Okay. So I using the one of the example is Peyer's patches. So you will see this diagram. So normally Peyer's patches is you can found in the small intestine. So in the small small intestine, near the small intestine. So normally you will see this structure. So this is the small intestine structure, and then this is the places that we call as Peyer's patch. So there are many lymphoid tissue at there. So the structure of GALT is well described and range from loose, barely organized cluster of lymphoid cells in the lamina propria intestinal villi to well organized structure. And then normally its function is to large population of antibody producing plasma cell and boost number exceed that the plasma cell in the spleen, lymph node, and bone marrow combined. And then next, this is the development of B cell and T cell. So as I mentioned before, B cell and T cell is produced in bone marrow, right? But only B cell is major in bone marrow. After that, T cell will be transported into the thymus for maturation. So this is very important, you need to know. Okay, this is the summary process, overall process of the blood cell, not only B lymphocyte but at all the blood cell. As I mentioned before that we have the stem cell we call as hematopoietic stem cell, HSC. It is inside the bone marrow. It is as the stem cell. So normally this stem cell will be developed divided into 2 different precursor. Either it is myeloid precursor or lymphoid precursor. So in our syllabus we will more focus on lymphoid and then lymphoid also can produce natural killer cell, lymphocyte, and also dendritic cell. But in our syllabus we will more focus on lymphocyte. As I mentioned, T cell is developed in the thymus and B cell is developed in bone marrow Okay, and then we know that T cell can be divided into helper T cell, T4 cell, and also cytotoxic T cell, T8 cell, right? Okay. So this is a summary. So about the development of lymphocytes, so as I mentioned, HSC is the origin of the blood cell will differentiate into major blood cell by hematopoiesis. So under condition when your immune system is required higher amount of blood cell, so your HSC will be stimulated and undergo hematopoiesis to differentiate more blood cell, and then when your HSC is induced to differentiate and then lose its cell delivery capacity and make more both lineage commitment trace. So either it can fall lymphoid progenitor CLP, or it can form the myeloid progenitor MP. You need to know. Okay, now we go to development of T cell. Okay, so this is the summary process of the development of T cell that you should know, you should learn Okay, so normally development of T cell, I already mentioned, it will include HSC. After that, it will go to the, it will develop into the lymphoid progenitor. Okay, after that it will migrate migrate transfer from bone marrow into the thymus. So during the thymus, it will process the T cell development lah. So actually it is very complicated process, but in your syllabus you don't need to know each process lah. You just need to know the overall process only. So once the T cell precursor, the T cell, the immature T cell that from the bone marrow enter the thymus, it will undergoes the first stage we call as early thermocyte development. Where the cell is lack of the CD4 and CD8 protein. So CD4 and CD8 protein, I think everyone should know, right? It is play the key role that differentiate the helper T cell and also cytotoxic T cell, right? So they will have a CD4 and CD8. So at this stage, it need to rearrange. The T cell need to rearrange their gene to produce, to express the CD4 and CD8 receptor. So after the thermocyte development is developed, it will produce the double positive CD4 and CD8 cell. So meaning that this cell will contain CD4 protein and CD8 protein. Okay. However, in our as you guys know that in T cell we containing helper T cell and also cytotoxic T cell, right? And helper T cell contains CD4 protein, and then cytotoxic T cell contains CD8 protein. So normally they need to select whether it want to go to CD4 or CD8. So it will using the positive selection. So actually got positive and negative lah. So if it is negative selection, it will die. It will go to other type of T cell. But if it is positive selection, it will either choose it is CD4 protein or CD8 protein. After that, when they choose CD4 and CD8 protein, they will go to another negative selection, meaning that this receptor need to bind with the MHC complex, which is intermediate binding, cannot fully binding, because it need to know that which cell is self, which cell is non-self. So all the MHC complex in our body will be detected as self. So they can intermediate binding only. If they fully bind, meaning that it is considered positive selection. So this kind of cell we cannot use. Because if positive binding totally bind, meaning that it is considered that the cell will recognize that this is non-self, and then it will activate the immune response. So we don't want this. They will detect that intermediate binding, so that they will consider as the cell itself. This cell should be adhered, so that the cell will not activate the immune response. So once they undergo the negative selection, now the cell is already activated, already be mature, and then this T lymphocyte will be migrate into the blood again, and then go to the lymphoid organ, other lymphoid organ, go to the upper lymphoid organ, especially lymph node, for the adaptive immune response, for the activation. So it will activate the adaptive immune response, cell-mediated immune response. Okay. And then this is about the explanation. So yeah, you can read yourself. Okay. And then about the development of B cell. So about the B cell is different from the T cell. So normally the B cell as the what happening in T cell. So normally the stem cell in B cell already produced. It will become the lymphoid progenitor. So some of the lymphoid progenitor will remove to the thymus for maturation of T cell. The remaining one will develop into B cell. So the develop of B cell will develop into this further stage by regulating the gene by regulating the gene of the B inside the B cell, so that it can produce the antibody lah. So normally it can go to the pro B stage, pre-B stage, immature B stage, and also major B stage. So after it go to major B stage, it will release from the bone marrow and then go to for the humoral immune response. Okay, so this is about the explanation. So normally as differentiation proceed, developing B cell expand on each cell surface, and then some of the signal from the receptor will induce a differentiation. Okay, next we go to the antibody structure. So normally in antibody, so this is the antibody. Actually this is not the fully completed antibody lah. So actually this is the B cell receptor. This is the B cell receptor that's still attached to the B cell. So for antibody, actually it is already released from B cell, but actually the B cell receptor and the antibody structure is totally the same lah, just that this one is still attached in the B cell, but when it is detached from the B cell, it will become the antibody. So normally this structure is like this. So normally antibody is Y-shaped protein that synthesis from B cell, and then it contains 4 polypeptide chains. So you see here 1, 2, 3, and 4 polypeptide chain, and then they will contain 2 heavy chain and also 2 light chain, and then each heavy chain contain light chain. Each heavy and light chain contains constant region and also variable region. So this is the light chain, and then this one that attached to the B cell is called as heavy chain, and then each chain will contain variable region and also constant region. So normally what is the meaning of variable and constant? So we know that these are the polypeptide, right? So they are made from amino acid. So constant region is that all of the antibody will have the similar amino acid sequence. So in the constant region, all type of antibody, whatever type of antibody that you know that we have 5 type of antibody like IgG, IgA, IgE, IgA, IgD, all that in the constant region, all the amino acid sequence are same, but the difference is the variable. So already mentioned variable. So each different type of antibody will have different type of amino sequence. So this is the key region that containing antigen binding site. So this variable is highly specific. They only can bind with the certain antigen only. So when this variable is only recognize A antigen, so only A antigen can bind for it. B antigen cannot. And then the region, between the region for the constant region are bonded by the disulfide bridge. Okay. And then as I mentioned, we have 5 major type of antibody, right? IgM, IgD, IgG, IgA, and IgE. Okay, so first type of the antibody is IgM. So the IgM structure is look like this. So actually it is the pentamer structure. What is the pentamer structure? Because it contains 5 structure of amino acid. So this one is we say as monomer structure. So it is pentamer structure. It consists of 5 unit of antibody. 5 basic unit of antibody lah, I can say 5 basic unit of antibody. So it look like pentamer. So normally the main function is that it is the first antibody produced in humoral immune response, meaning that in humoral immune response, normally the antibody produced is this. They will produce this antibody first. After that, they will switch. Actually, they are the mechanism switching, but in our syllabus, you don't need to learn it. So actually, they are the switching process. They will switch the IgM to other type of antibody lah. So normally the function is all the function lah. Neutralization, agglutination, complement, activation is all the function of IgM. And then another one is IgD. So IgD is the for the function for the antibody that play the critical role in maturation of B cell, and it help to activate B cell following antigen binding. So it is the monomer structure. And another one is IgG. So IgG is the spatial antibody, is also monomer structure, but it is a very special antibody that only the antibody can pass through the placenta. So meaning that the antibody that produced from mother, it will be passed through to the placenta to protect the fetus. So this antibody is to protect the fetus. So normally it will function as neutralization, all these function, but the key function is to protect the placenta, because it is the only antibody that can pass through the placenta only. Another one is IgA. So this is structure IgA. So IgA is the dimer structure. So normally this is the structure that normally is present in the epithelial surface. All the epithelial surface, like the mucus, or in your kidney, all the epithelial tissue surface, it will IgA will be appear. So normally it is act as the prevent the virus and prevent the bacteria to attach this epithelial surface lah. Okay, and then the last one is IgE. So IgE is the activation of the basophil and mast cell that normally is play the role in against parasite and allergy, and also including inflammation. So so it can bind to the mast cell that contain histamine that trigger many allergic symptoms and also parasite symptom. Okay, and then it is a monomer structure. Okay, so that's all for the 5 class of antibody. Okay, now we go to the development of anti-immunity, which is adaptive immune response, which is very important in this chapter. Okay, so in the adaptive immune response, it is already say it is a third defense system. So T lymphocyte and the B lymphocyte play the role in the adaptive immune response, right? And then we know that there are two types of immune response. First one is cell-mediated immune response, and another one is humoral immune response, right? That you guys know. So if you want to activate the cell-mediated immune response and also humoral immune response, you need to activate first. So you need to activate helper T cell first. Helper T cells play the role in activate the adaptive immune response. If no, the adaptive immune response cannot activate. So helper T cell, you need to activate your helper T cell first. So how to activate helper T cell? So helper T cell play the role in activation of adaptive immunity. So normally in helper T cell, before the helper T cell activate, want to activate helper T cell, the macrophage that act as the antigen presenting cell that we know, then they will engulf the pathogen first. Well, engulf the antigen or engulf the pathogen first. So normally before that, the foreign molecule must be presented that bind to the antigen receptor or helper T cell, and then the APC must display the antigen. So normally they need to the macrophage or dendritic cell act as the antigen presenting cell. They will phagocyte the pathogen first, and then they will hydrolyze the antigen, and then to form the antigen. And then this antigen will bind to the MHC2 molecule. MHC2 molecule. And then this MHC2 molecule, MHC molecule will displace on the surface of the macrophage. So that's why that now macrophage now acts as the antigen presenting cell. So it will displace the antigen on the surface of the macrophage. So after that, once the antigen macrophage already display the antigen on the surface of the macrophage based on the Cas2 MHC molecule complex. So normally the Cas2 molecule will provide a molecular signal, and then the SPC will display antigen fragment complex with Cas2 MHC, and then this specific antigen receptor receptor of the helper T cell, they are the receptor, right? And helper T cell contain the antigen receptor, will start to bind toward the antigen that displayed by the APC, and then there are also the CD4 accessory protein. CD4 accessory protein will also bind to the Cas2 MHC molecule to keep the cell joined together. So the T cell receptor, antigen receptor, will bind to the antigen, and then this CD4 accessory protein will bind to the MHC2 molecule. And then once they bind, the APC was starting to release the cytokine, which is the interleukin 1 to activate the helper T cell. So now your helper T cell is now activated. So this helper T cell is activated. It was starting to release cytokine interleukin 2 for proliferation. So meaning that it will proliferate, divided to produce more and more activated helper T cell. And then this helper T cell can start to activate the either humoral immunity immune response or cell mediated immune response. Okay, but for now it is just complex for the same antigen only. So what antigen that activate helper T cell, this response only can respond to that kind of antigen only. That A antigen, then it only can respond for A antigen only. And then about the next is about the cell mediated immune response. So I already mentioned, when you already activate the helper T cell, so it already activate helper T cell, it will release interleukin 2 to activate cytotoxic T cell. So your pathogen, so for the cell mediated immune response, the cytotoxic T cell play the role. So only cytotoxic T cell will directly digest the cell that already infected by the pathogen. So how it digests? So when the cell is already infected by pathogen, this is a normal cell already infected by pathogen, it was starting to display the antigen fragment. Antigen fragment that will react with the class 1 MHC molecule. So similar to activation of helper T cell, but the difference is the helper T cell is bind to the antigen is bind to class 2 MHC, but for this one, the infected cell, it will bind to the class 1 MHC. So that when bind to the class 1 MHC, it will form the complex class 1 MHC antigen complex to displace the antigen on the surface of the infected cell. So this activated cytotoxicity cell will bind toward the T cell receptor of the cytotoxicity cell, will bind toward the antigen that display on the infected cell, and then there are also the CD8 accessory protein will bind toward the class 1 MHC molecule. So once they already bind, the cytotoxicity cell was starting to release two components, perforin and also granzyme. Okay, so perforin is to make the pore to the infected cell, make the pore on the plasma membrane of the infected cell, so that when there are the pore, so that the water molecule will be rushed in flux into the cell, cause the cell lysis. So it will kill the cell lah. Not only that, it will also release the granzyme, a type of enzyme that can break down the antigen cell by endocytosis. And then this enzyme will initiate apoptosis within the infected cell, resulting in the fragmentation of the nucleus and cytoplasm, and finally it will die lah. And then so this is a cell mediated. So why he say that it is cell mediated? Because it directly kill the cell. So the cytotoxic T cell play the role. Okay, another one is humoral immune response. Humoral immune response is the part of that using B cell. So as I mentioned, helper T cell need to be activated first, then you can proceed to humoral immune response. If no humoral immune response cannot be proceed. So now B cell will play the major role in the humoral immune response. So B cell is activated by the activated helper T cell, and the binding of B cell with the antigen which have the same epitope with the helper T cell. So as well as the helper T cell is activated, your B cell also will bind to the same antigen that bind in the helper T cell using B cell with antigen receptor independently, meaning that your B at the same time when your helper T cell is activated, at the same time your B cell require to display the foreign antigen, and then this antigen is similar to the antigen that activated by in the TH. So for example, when your activated T cell is A antigen, then your B cell is need to bind to A antigen too. So that it will display the antigen fragment. It display the antigen on the antigen receptor, antigen binding site of the B cell receptor. Now the B cell will act as the antigen presenting cell, where it will display the antigen fragment with class 2 MHC molecule on the cell surface. So it will display the antigen fragment, and then this helper T cell will bind to this B cell. And then it will bind to this B cell using the CD4 accessory protein 2. So CD4 accessory protein will bind to the MHC class 2 MHC, and then the binding receptor of the T cell bind to the antigen receptor of the B that display from the B cell. Now the B cell is activated. So once the B cell is already activated, it will proliferate into two kind of cell. Either it will proliferate into memory B cell or plasma cell. So when it is proliferate into the plasma cell will starting to secrete the antibody. I will start to synthesize the antibody lah. So this antibody are only highly specific to the A antigen only. And then this is the overall process. So normally, when you have the antigen in the cell, it will taken by antigen presenting cell. And then when your antigen presenting cell, you need to activate the helper T cell. So once the helper T cell is activated, it will go to the cytotoxic T cell. It will activate cytotoxic T cell, and then cytotoxic T cell will be activated to become memory cytotoxic T cell or active cytotoxic T cell. And then this active cytotoxic cell will undergo cell-mediated immune response, which it will directly kill the infected cell lah. But when the helper T cell in humoral immune response, it will activate B cell, and then antigen will also bind to the B cell receptor, and then this B cell will proliferate into plasma cell and memory B cell, and then plasma cell will starting to secrete antibody. And then this memory B cell and memory cytotoxic T cell is already say is memory, right? So when there are the second exposure, second infection by using the same antigen, this memory cell will be activated, become plasma cell, or totally it will become the anti-cytotoxicity cell. It will directly go to the to proceed the activation. So it will not don't need to have an activation again directly, because it already have memory. Your body already have memory, or already know this one is antigen, same antigen, so it will directly go to the stimulate the human immune response. It will don't need to go through the original process again. It will go to the original process again unless your antigen is different antigen, new antigen exposed. Okay. Okay, now we go to the antigen antibody reaction. So this is special for human immune response that will produce antibody. So how the antibody work? So in our syllabus we will learn about neutralization, agglutination, precipitation, and also complement activation. So normally neutralization, agglutination, precipitation, the final result is to lead to phagocytosis by macrophage. But for the activation complement system, is lead to cell lysis. Okay, so we look to one to one. So normally neutralization already say neutralize, meaning it is to neutralize the pathogen. So normally the antigen will bind to the antibody will bind to the antigen to prevent the pathogen from ever initiating an infection. Is known as neutralization, meaning that this is the process before the antigen, before the pathogen infect the cell, then the antibody will take action already. So this is to prevent cell-mediated immune response. So before cell-mediated immune response is already occur, the cell is already infected. But this one, your pathogen, before your pathogen infect the cell, the antibody already take action. So this one is we call as neutralization. So normally will bind to the pathogen or a toxin that interact with hospital, masking them and inhibit them to enter the. So it will inhibit them. It will not directly kill. Antibody will not directly kill the pathogen. You need to know that. You cannot say that antibody will kill the pathogen. No, antibody just act as the dia macam worker je to collect the antibody, to collect all the antigen or all the pathogen in the one way, and then directly prevent them to infect the cells only. So that after that when the antigen when they already bind to the antigen, they will prevent the pathogen to bind and infect the whole cell. After that, after a long time, they will be phagocytized by macrophage. And then next one is agglutination. Okay, agglutination is bind to the epitope of more bacteria simultaneously to the pathogen. So meaning that one antibody, they will bind toward many bacteria. Not only one bacteria, but for this one, this is only bind only one bacteria only. But for this one antibody can bind many bacteria. And then they can clamp together. They will aggregate together. And dia macam collect all the bacteria and aggregate to one place. So to inactivate the cross-linked bacteria, so that after that this complex, all the large complex will be engulfed by the macrophage for phagocytosis. And next is precipitation. Okay, precipitation is look like similar to agglutination, but different is precipitation is to bind the antigen only. This one agglutination is to bind the epitope on the bacteria. For this one is bind to the antigen, the free antigen that survive on your blood at the any space. So it will bind to the antigen, it will precipitate it, become the insoluble substance, and then this substance will be phagocytized by macrophage. Okay, next is about the complement fixation. process antibody are work together with protein of complement system, meaning that your antibody will bind toward the complement protein. This is not free. This one, if you compare to the this one is antibody is free at all. If free at all, when it find the antigen, then it directly bind. But for this one is that the antibody only bind toward the cells is already infected. So the antibody will bind to the antigen on the surface of foreign cell. So the cells that already infected by the pathogen will be using this complement fixation. But for this one is that the cell, the pathogen not yet infected. The pathogen is freely moving around in your blood vessel or in the lymphatic vessel. But for this one is that your cells is already infected already. So when your cell is already infected by the pathogen, so that the antibody will cooperate with the complement protein, and then your antibody will bind to the antigen. Okay, this will trigger the formation of membrane attack complex. So it will trigger the formation of membrane attack complex at here. So this already say that membrane attack complex, meaning that it's attack your plasma membrane. So it will form this membrane attack complex will bind toward the plasma membrane, and then it will form the hole on your plasma membrane. Hence, all the water and the ion will influx into the cells, and then this will cause the cell becomes lysis. And then this will factor that promote inflammation and can stimulate phagocytosis. We go to the concept self and non-self. Okay, concept self and non-self. So this one is especially very important. Okay, that can and active inhibition of the immune response against self-protein. So normally MHC that you see just now, MHC is the protein that play the role in recognizing self and non-self. So we have two class of MHC, right? MHC class 1 and MHC class 2. And then we know that MHC class 1 is only special that will be bind toward by the CD8 protein only, which is cytotoxic T cell. The cytotoxic T cell contains CD8 protein, right? And then it will directly bind to MHC class 1 to activate the cell-mediated immune response. And then MHC class 2 is that it contains CD4 as a solid protein that is present in the helper T cell that will bind toward the MHC class 2 to activate the helper T cell and also activate the humoral immune response. This is what you should know lah. Okay. So we know that MHC class 1 is expressed as glycoprotein in all of the cells, meaning that actually our cells, all of our cells, not only lymphocyte, all of our cell, normal cell, will have MHC class 1. So it play the role in the cytotoxicity cell only. So meaning that activation is meaning that all of the cell containing MHC class 1. And then MHC class 2, I already mentioned MHC class 2 is the expressed glycoprotein on the surface of APC antigen presenting cell only that play the role in presentation of peptide antigen to helper T cell. And then MHC play the role in vagonization of self and non-cell, as MHC consists of non-classical region which we nucleate for every different individual. So every different individual will have different sequence in MHC protein. So like me, I have A type of MHC class 1 and MHC class 2, but like you, my friends, or even my mother, they have different MHC class 1 and MHC class 2 because they are the non-classical region. So you will see here this region. This is the genetic consequence of the MHC that will be expressed MHC class 1, class 2. So this is the class 1, this is the class 2. Class 3 you don't need to learn. Okay. So normally this is the gene, right? This is the gene, and then this one it will express MHC class 1. This one is the place it will express MHC class 2. So normally you will see that the black color one is the non-classical region, meaning that different individual will have different of the region, different amino acid lah. So that is why the MHC play the role in the recognized classical. So although in organ transplant, so that is why that you need to relate into organ transplant. So maybe I am sick. I have a kidney failure. I need to have a kidney. So not all the kidney from any people can accept. I can accept. I need to do the MHC compatible first. So only the MHC compatible first. Why? Because I already mentioned different individual will have different individual have a different sequence of the MHC class. So although it is similar to MHC class 2 or MHC class 1, but since your helper T cell, it look like MHC class 2, but when the helper T cell is go to helper T cell, check, tak sama, because your sequence is not same, they will assume that it is pathogen, and then it will attack you. I will attack you. So when the MHC compatible is incompatible, I still accept your kidney. Then your helper T cell will assume that your T cell will assume that your pathogen, your cell is pathogen, because APC is directly assume that your cell is pathogen already. The kidney, your recipient kidney is pathogen. So directly helper T cell will directly recognize MHC class 2. Whether this MHC class 2 is yours one or others one, and then they detect is others one, and then it will activate the immune response, and then you attack your kidney lah. So this one is we call as rejection occur. Okay, so that is why that you need to relate to the organ transplant. So normally organ transplant, we have 4 types of organ transplant, right? Autograft, isograft, allograft, and xenograft. So normally autograft is that will transfer your tissue from other part from one part to another part, but if same person, like skin transfer lah. Like your skin is injured like burning process. Normally your doctor will take your skin from leg, transfer to your hand. Maybe in your hand you are injured in burn, and then you need to do a skin transfer, and then doctor will take your skin from your leg and then transfer to your hand. So this one is we call as autograft. And then isograft is normally tissue transfer is genetically identical individual. So normally this one is only occur in identical twin only. And then this one is allograft. Allograft is that transfer from different individual but in the same species like human lah. We transfer organ from human to human. Xenograft is the new technology that the tissue organ transplant is different species. Like for nowadays, that the American research, they do research about that to transfer the organ from pig into human. Kidney from pig into human, and also kidney from and also heart from pig into human. This is we call as xenograft. Okay, now we go to rejection. So normally autograft and isograft no rejection. Normally will not have a no rejection. Why? Because autograft is using your own tissue, so no rejection at all because your MAC is totally same. And then isograft also no rejection because you are using the identical twin, meaning that all of your genetic diversity is totally same as well. So isograft is also no rejection. Only allograft and xenograft will provide rejection. It compulsory provide rejection. So normally you will see this diagram. So normally for the this autograft acceptance, they will accept lah. But you will see that in allograft they will have the double type of rejection: first set rejection and second set rejection. So in allograft you will have a rejection. So normally you will see that during the rejection process, you will see that your blood cell was starting to attack your tissue, and then you see that in your tissue will become necrotic tissue, meaning that this tissue is already died, and then it will cause many infection and inflammation as well. So this is what we call as rejection. Okay, so this one is tissue that have the sufficient antigenic similarity, allowing transfer without immunological rejection, are said to be his compatible. So this only occur in identical twin only. And then when the display significant antigenic difference, so this one antigenic difference is we call as MHC class. So MHC class I already mentioned is displayed on all type of the cell. MHC class 2 is class 1 is displayed on the every type of any cell. So when there are the antigenic difference, meaning your MHC class 1 is differ and some differences, it is said to be his compatible, meaning that there are the immune response lead to tissue rejection, and then this will happen in allograft and also xenograft. And then we know about the rejection mechanism. So normally rejection mechanism can be separated into two phases. So normally graft rejection occur in stage and can be caused by both humoral and cell-mediated response to the allo antigen MHC. So for now, your NHC become antigen. Your MHC become antigen now, expressed on the cell of the graft. So the recipient organ like the kidney, the recipient organ now will express the MHC, right? And MHC. So this we call as allo antigen now. Your MHC now is becoming antigen. So if your MHC is incompatible to the recipient, the donor MHC is incompatible to the recipient, then rejection will be occur. So there will become two phases: sensitization phase and also effector phase. So sensitization phase normally is occur after transplantation. It will occur directly, and then normally your lymphocyte was starting to proliferate. They were starting to detect that this your donor, the organ of donor is as the non-self, it was starting to activate the immune response. So if fetal stage is already activate immune response, your B cell and your T cell is already activated, it will start to attack the organ. It will start to attack the organ already. It will destroy the organ. So that is why the fetal stage. So in sensitization phase, your T lymphocyte, your macrophage was starting to engulf your organ engulf your donor organ, and then your donor organ, your macrophage will act as the antigen presenting cell, right? APC. So it will proceed to this process lah. Okay, next is about the HIV infection. Okay, so HIV, I think everyone should know HIV infection will lead to AIDS virus. So HIV is one type of virus. So this is the structure of virus. So if you see this virus, normally this virus is containing RNA virus. It only containing RNA viral genome, and then this yellow color one is capsid. And then this is the envelope virus. This one is our envelope, and then this one is our glycoprotein. Okay, we need to know the structure. This one, the blue color one is viral RNA, yellow color one is capsid that protect the virus, protect the RNA viral genome. And then this one, the purple color one, is we call as envelope. And then this one, light blue color one, is we call as glycoprotein. You need to know the structure of HIV, because maybe in structural question it can become out. And then the infection of the HIV. So normally HIV is very smart virus, because it can directly attack your helper T cell. It direct attack helper T cell one. So CD4 on target cell. Why? Because it will bind toward the CD4 accessory protein. So which cell contains CD4 accessory protein? Helper T cell, right? So it will directly bind toward your CD4 accessory protein. And then once it bind your HIV virus will really bind to a chemokine receptor, and then fuse to the target cell membrane, and then it was starting to release viral RNA genome. And then this viral RNA genome, single-stranded RNA genome, were using the reverse transcriptase enzyme, triggers enzyme that produced from HIV virus, because reverse transcriptase enzyme in our human, we don't have this enzyme. So this virus have this enzyme. It was starting to produce double-stranded RNA DNA hybrid. So will catalyze reverse transcription of single-stranded RNA, forming RNA DNA hybrid. And then the original RNA template is particularly degraded by ribonuclease, followed by synthesis of the second DNA strand. So it was starting to form HIV double-stranded DNA. So once it already formed the double-stranded DNA, it will translocate into the virus, into the nucleus of the T cell by the integrase enzyme. So once the already integrated translocate into the nucleus, it was starting to integrate our DNA. So meaning that transcription receptor will stimulate the transcription of both viral DNA into genomic RNA. So meaning that this one is already DNA. When it's translocated into our DNA, meaning that it will be considered as one type of DNA. So when it is considered one type of DNA, what will happen? Gene expression will happen, right? You know that gene expression happen. So transcription process and translation process will happen. So transcription transcription process will produce mRNA. So this mRNA already is the HIV mRNA, and then this mRNA will be diffused out from the cytoplasm, and then translation happen. And then when the translation happen, it will produce the HIV protein. So these are all the protein is the component to produce the HIV virus. And then this mRNA also, this single-stranded mRNA that produced from the transcription also can be diffused out from the nucleus, and then become the new viral RNA to the new HIV virus, so that all of the protein HIV and the protein will become assembled in your helper T cell to form a new HIV virus, and then your membrane will back off to forming the viral envelope, and then new HIV virus will be produced, and then this virus will infect another helper T cell, infect again and again, and then this one is more summary. So that is why that patient that get HIV virus is not die because of HIV. They die because of weak immune response, because it already attack your immune response, it already attack helper T cell. When your helper T cell is attacked, meaning no adaptive immune response. So meaning that when there are a serious pathogen, severe virus like COVID-19 like that, because due to don't have the helper T cell, your helper T cell is very less, because it already attacked by the HIV. Now you don't have the helper T cell, then you cannot activate immunity, adaptive immunity response. So when the COVID-19 virus attack you, then you will directly die lah. Your risk to die is very high. Okay, next is the SLE, last topic. SLE about the immune disorder, systemic lupus erythematosus. So our syllabus we just learn about this kind of immune disorder only, only one type. So we call as SLE. So what is SLE? Normally this SLE is the chronic inflammation occur when your body tissue are attacked by its own immune system, meaning that your immune system detect your body tissue is non-self, so be your body tissue is self, but now if we detect that your body tissue is non-self, it attack your body tissue. So if SLE can produce the abnormal antibody, this antibody will attack your target tissue rather than for an infection agent. Should be your antibody should attack infection agent for an infection agent, but now no, your antibody attack your target tissue pula. Then immune response, when immune complex or antibody with various nuclear antigen are deposited along the wall of small blood vessel, type 3 hypersensitivity reaction will occur. So this type 3 hypersensitivity will activate the complement system, generate membrane attack complex to damage your blood vessels wall, resulting in vasculitis and glomerular nephritis. And also, if the complement activation is still enveloped again, it will elevate your serum level of certain complement fragment, leading to neutrophil aggregation and attachment of the vascular endothelium. And over time, your circulating neutrophil will be declined, and the occlusion of various small vessels will develop, which can lead to widespread of the tissue damage. And then this is about the symptom that will be happening in HLE patients. So normally swollen joint, muscle pain, and then your fever cannot be explained. Although you are the healthy person, but you cannot. Your fever can come suddenly one, and then you have rashes like the red point in your face, and then chest pain, and then your hair is loose, and then your purple finger, you have a purple finger, and then sensitivity to sun. This patient cannot expose to the sun one, and then the swelling in leg, oedema, you will have the oedema lah, and then mouth aerosol, swallow gland, and also fast-stream fatigue. Okay, so that's all for this chapter lah, chapter immunity. Okay, so now it's already 11:45. Okay, 11:45. So we will take a break, take a lunch time, and then we will proceed to the class again. Proceed to the class again 2:30. I will finish up the last chapter lah. I will finish up the last chapter. Okay, so we will see you at 2:30 lah. Again, proceed to the class again 2:30. I will finish up the. Okay, so we see you and take a lunch. So goodbye, and we will see you again at 2:30. Okay, so now it's already 2:30. Okay, so we will continue our revision class. So for today, we will now I will continue to the last chapter lah, infection disease, chapter 13, and this is the last chapter for semester 2, right? And then in this chapter, you have 6 topic you need to learn, right? The first is about the introduction of infection disease. You need to know what is the meaning of infection disease, what is the meaning of infection, what is the meaning of infection agent, all this. And then next you will learn about the type of infection disease, right? So we are told to get in our syllabus. We have 4 type of infection disease you need to learn: dengue, dengue, cholera, tuberculosis, and also malaria, right? So this is in our syllabus, but actually in real world there are many type of infection disease lah. So including the now as popular COVID-19. COVID-19 also is considered as one type of the infection disease too. But however in our syllabus we only learn the 4 type that I just mentioned just now lah. Okay, so we are wasting our time. So this topic actually is very short topic, I can say. One of the very short topic similar to the chapter 7 lah. Okay, so now we go to the definition of infection. So you need to know what is the meaning of infection. So normally the infection is that it is the process where the microbe or parasite or pathogen enter into your body. So that when they enter into the body, this one is recalled as infection. And normally the infection can be begin in anywhere in anybody. So any part of the body, if there are cells, once the body have cells, then it can cause any infection. And then this infection can maybe spread out through the surrounding. So the infection not only is at a certain place, it also can be spread out into another places. And then infection normally will cause fever and other health problem depending where the infection is occur lah. And then normally when our body immune system is very strong, normally we can fight with the infection one to treat our infection. But if our body immune system is very weak, then infection maybe can cause very serious problem. Okay, this one definition of about the infection disease. This is very famous question that will be asked in essay question. Already asked many times. I'm not mistaken, later we will see it. After this class I will do analysis, right? If not mistaken, already 4 times come out, 4 times already, 3 marks. What is the meaning of infection disease? So it already come out many times, so whatever, maybe it will come out this time or 2. So you need to remember lah. Kalau if come out, then you are very lucky lah, because the definition, it contains 3 keywords. The first keyword is infection disease is caused by pathogens. See, this is the first keyword. And then they are transmitted by a vector. Second keyword. Third marker is it will be transmitted from an infected person to an uninfected person. This is the third keyword. If you write all this, then you will get full marks, 3 marks. So you need to very important. First keyword is it is caused by pathogen. You cannot say it is caused by vector. Cannot, no. It is caused by pathogen and it is transmitted through a vector. And then this transmission can also cause from an infected person to an uninfected person. So these are the 3 keywords you need to remember lah. And then about the infection agent. So normally the infection agent normally is by pathogen lah. So this is the example of the transmission infection agent. So normally the infection agent can be virus, can be bacteria, can be fungi, can be protista. Protista. So normally in our syllabus we know that we learn about tuberculosis, right? So the tuberculosis, the pathogen used is bacteria, and then the cholera is also bacteria, and malaria and dengue. Malaria is protista. Actually, dengue is virus, and then the transmission vector can be through the air, like COVID-19 is through air, right? Airborne disease, airborne transmission, and also some of them is through the food, and some of them is through the animal, like mosquito. Okay. First we go to the dengue. So dengue, I think this disease is very famous, right, in Malaysia. So maybe some of you maybe have said for dengue, but some of you not. So dengue actually is a famous infection disease in Malaysia lah. Not only in Malaysia, and also especially in tropical country. So normally dengue is an acute arthropod-borne viral infection that plays a very socio-economic and disease burden on many tropical and subtropical regions. Okay, so the causes of dengue. The infection agent of the dengue is dengue virus, DENV. So there are 4 types of dengue virus. Then 1, 2, 3, 4. So there are 4 different types of dengue virus, and then the scientific name of the virus is we call as flavivirus. Dengue virus is the general name. If the examination want you to come out, you need to write the flavivirus, not dengue virus. Dengue virus is just a general name only. And then the transmission vector through this dengue virus is Aedes mosquito, Aedes mosquito, right? So there are many species actually under many species, but Aedes aegypti is the more aegypti, and also Apophysetus is the most species that transmit the dengue virus, and only female mosquito only. Male mosquito does not transmit the virus. So this is the mosquito lah, and then this is the structure of the dengue virus. Okay, so in this chapter, all the disease you need to know what? You need to know transmission, right? You need to know cause, what caused this disease. You need to know transmission of the disease, and then you need to know prevention of this disease, right? So okay, so you already know the cause. The cause of the disease is through these 2 substance, the dengue virus and also vector. Okay, so when the patient, so what of the cause is when the female infected dengue, infected mosquito that carry the flavivirus, they will bite you. After they bite you, what will happen? This is the transmission method lah. When they bite you, what is your happen? So then normally the dengue virus will infect your white blood cell, and then multiply at day, and then it will cause your spleen and lymph node become swollen, because it already infect your white blood cell. And then we know that our lymphatic organ like lymph node and spleen are the major places to major location to play the role in adaptive immune response, right? Since it already attack your white blood cell, then your spleen and lymph node also will become swollen. Okay, so after they bite you, they will have 2 types of infection will be occur: primary infection or secondary infection. So what are their difference? Primary infection is that if you are the first time infected with dengue. Okay, as you guys know, dengue virus contain 4 types, right? Any types of virus that you first time infected with dengue, either type 1, type 2, type 3, type 4, anyway, if you first time infected with dengue, then you will consider as primary infection. However, if you are already infected with dengue, but you infected again, you already be treated, you already sembuh, already become healthy, but maybe several years ago, or maybe 1 year, 2 years ago, you still infected by dengue again. and then this infection, your dengue virus is differ from your first time infected. So maybe first time infected, your dengue virus is dengue type 2, but now the second time you infected is type 3 virus. So totally different. So if the virus that you infected is different, you will cause secondary infection. Okay, so what will happen in the infection? So normally when a people already infect dengue virus, your immune system will fight with the virus so similar what you happening in the adaptive immune response that I explained just now. But the most important thing is, in dengue infection, if the person is infected with another type of dengue virus which is different from the first time, that person will maybe get the worst infection than the first time. This is different from the adaptive immune response that we learned just now, because I already said adaptive immune response contain memory cell, right? So after you go to infected second time, your immune response should protect you. But for dengue, they not protect you. The immune response not protect you. But they will give you a worse infection again. Why this happen? Because of ADE, antibody independent enhancement process. So normally ADE infection will occur when the pre-existing antibody from the primary infection dengue virus will bind to the dengue virus, the different serotype, different type, resulting the antibody cannot neutralize the virus. So I already mentioned dengue virus contain 4 types. Although there are 4 types, but normally the structure of the virus looks similar. The difference is just the genetic sequence are different only, but the virus structure is totally similar. If you just see the surface, you cannot differentiate them. So of course your white blood cell, your B cell, your antibody also cannot differentiate them. They will assume that this virus is similar to the first time that you infected. They will bind to it. However, once they bind, they cannot neutralize, because this is totally different virus. Although it looks similar, but actually the antibody can bind it, but it cannot neutralize it. It can bind it, but it cannot neutralize it. So what will happen is this virus will make them bind toward the Fc receptor of the host cells. Because we know that antibody neutralize it. After that, the neutralization is to prevent the pathogen enter the host cell, right? But for now, it cannot neutralize. So so the pathogen still can enter the host cells. So it will further infect the host cells. So ADE make the virus more easy to enter the cell, because now pathogen containing the antibody. The antibody make the virus easy to infect the cells compared to no antibody, right? You got antibody, later it will go phagocytosis, right? So it already help the virus, like the antibody should be prevent the virus enter to the host cell. But now your antibody already become a become a car to make your virus easier to infect the cells. Okay, so this is the infection of dengue. So you know that in primary response, your dengue is directly infected your white blood cell one. But for the secondary response, as I mentioned, the antibody is already bind to the dengue virus. So this antibody will bind to the receptor. So it makes the virus more easily to enter the cell, and then it will infect it, and then it will replicate more and more dengue virus. Okay. So normally it will cause the resulting of the infection will cause these two consequences. Classical dengue fever or dengue hemophagic fever. So classical dengue fever is the normal fever only. Normal people that you infected dengue, you will get this kind of fever one. But if the patient is getting secondary infection, it will get the dengue hemorrhagic fever, which is more serious compared to classical dengue fever. Okay, so normally what you will happen in classical dengue fever is you will have sudden high fever, headache, and then joint stiffness. What the scientific name is harvigia, myalgia, muscle ache, muscle pain, and then eating disorder. You don't want to eat, and abdominal pain, your stomach pain, and then you also have rash characterized by red area. Your skin will appear the flap of red area on your skin, and also inflammation will also occur on your mucous membrane, and also flushing also will occur. Okay, this is about the classical dengue fever. This is the common symptom lah. And normally this patient, if you are be treated very earlier, normally in 1 week or 2 weeks you will become healthy. But in DHF is very difficult. It's very difficult. Why? High fever and chills. Okay, in DHF they will have 3 phases: febrile phase, and then febrile phase, and febrile phase, and then another phase is vascular leaking phase, meaning this is more serious one. In febrile phase, normally you will get the symptoms similar to classical dengue fever, but in DHF, after you not only get this, you will get more serious symptom. So this is the more scientific lah, but actually you don't need to know. You just want to know that you will get the vascular leak syndrome, plasma leakage. Yeah, you will get the plasma leakage, and then you will bleeding. Bleeding may be occur, and then liver impairment and some of the organ impairment will also be occur. So liver impairment, cardiac impairment, CNS impairment, and your eye impairment may be occur also. So it is very serious, can cause very serious effect lah. Okay, about the prevention of dengue. So normally prevention of dengue is to prevent the transmission vector, because transmission vector, your Aedes mosquito is the key factor that transmit dengue, right? So if no this mosquito, then no dengue will be happen. So if you want to prevent them, you need to minimize the contact of the mosquito. So normally you can wear light white color and long sleeve, loosely colored and trouser to prevent the bite of mosquito. And also you can apply the pesticide like DEET, and then you can also install mosquito net to the door and window that mosquito cannot get into the indoor area. And then you need to remove any water container that can float with water, like flower pot, trays, and then bucket, jar, and your tire. All this is the places or breeding area for Aedes mosquito. You need to remove all this, and then also you need to dispose of osmotic waste that can accumulate the water. And then you need to always check and clean the drains, longkang, to ensure that the drain and the drain is the water is not blocked lah. Okay, and then you can also using the fish like the Gambusia amphinis and also Persilla reticula, the larva eating fish that can eat larva, and then to avoid doing outdoor activities. And also you can conduct the public awareness campaign to the society. Okay, next disease is about cholera. Next is about the disease of cholera. Okay, cholera is the major disease that normally caused by the water and by the bacteria and normally the people that drink the unwashed water lah. The water is already contaminated and they drink. So normally this disease normally is occur in the poor country. Normally is in the low and middle income country, especially low income country lah, or the country that now is in the war, dalam peperangan. So for the country is under the war, like the Palestine and also Ukraine, all these place, because that at the war country, your water will be contaminated, right? So contaminating water will have the possibility have the bacteria we call as vibrio cholera. So this is the major infection agent from the cholera. So vibrio cholera is the infection agent for this disease. So normally for this disease there are no transmission vector. No transmission. The transmission vector is the water. Normally it's contaminated food or contaminated water. For those who already drink the contaminated water, drink the contaminated water, unsafe water, unwashed food, unwashed vegetable, or uncooked seafood, all these all have the contamination. It is not clear. You will have the possibility to be infected by this bacteria called vibrio cholera. So normally it is result from consuming water that is already contaminated. Your water is already contaminated, and then if you drink this kind of water, or eating the contaminated food, or eating the food that washed by this kind of water, you will have the possibility to get this disease lah. So normally this disease is happening in poor sewage system, I already say, or unclean drinking water. So normally it's in the low income country, or the country is in the war lah. And then foodborne transmission may also occur when the individual eats the raw or undercooked shellfish. And then the bacteria also found in the sloughs in the river, especially a lot of backwater and the coastal water. Okay, so normally when this bacteria starting to enter your body, it was starting to infect your small intestine. So when it infect in your small intestine, it was starting to release the enterotoxin, one type of toxin called collagen. So it is an enzyme and collagen, and also enzyme protease, which act as the on the mucosal epithelium lining of the small intestine. So what are the effect of these two kind of enzyme? So normally this toxin will alter the surface protein on the epithelial cell in small intestine, and then they will become the receptor for the toxin of collagen. And then the collagen will stimulate the epithelial cell to secrete large quantity of chloride ion into the lumen of the small intestine. So this one, sodium ion and waste and water follow the loss of chloride ion. Okay, thus the water potential of the cell increase, and your total water potential gradient generated between the lining, small intestine and lumen. And then so the water will diffuse by osmosis out of the cell into the lumen. So normally this kind of patient you will vomit, and also like the severe dehydration. And then the symptom of this disease normally is abdominal cramp and severe dehydration, loss of fluid, vomit, you will vomit, and then dry mucous membrane, dry skin, and then you need to drink a lot of water. You will feel you are very thirsty, and sharpened eye, low urine output, leg cramp, and your blood pressure is undetectable, and wing hang and wing feet. And then the prevention of this disease is very easy and simple. The most easy prevention is to using the mineral water. You drink the safe drinking water only. If you drink the safe drinking water, then That's all. And also hygienic disposal of fees. Fees include proper sewage treatment, and then health education, like you need to wash your hand, eating the food before washing. Well, you need to wash your hand before eating, like that. And then also control of fire, which act as the vector in transferring facial material to food, and isolate the patient, and then carriers should be identified and prevented from working in the food industry. And then intermediate examination of diarrhea disease for bacterial content. So you need to check examination safety check. Okay. And then next is about the tuberculosis. Okay, so tuberculosis actually is about the lung disease lah. It is a chronic infection characterized by pulmonary infiltrate and the formation of granular with calcification, fibrosis, and cavitation. So normally the causes of this tuberculosis is caused by the bacteria. There are two bacteria will be caused, either Mycobacterium tuberculosis or Mycobacterium bovis. So it is caused by the Mycobacterium tuberculosis or Mycobacterium bovis. So normally, however, Mycobacterium bovis is normally found on the cow that emanated through the milk before consumption. So the fresh milk that directly come from cow may contain Mycobacterium bovis. This it can cause tuberculosis one. Okay, the bacteria will lose itself. So normally when you get this kind of disease, the bacteria will directly go into your lung. It will infect your lung, infect your alveolus, and then the bacteria will multiply in the macrophage, cause more nodula to be formed in the lung tissue, and then this tissue then will die and form cavities in the lung. So normally, pulmonary tuberculosis is the biological name when the bacteria attack to the lung due to the lockdown of defense mechanism. So now your lung is already be affected, and then if your bacteria starting to affect other organ, is known as primary TB infection. So normally our defense system can hold off the bacteria by rolling them within the lung. However, this bacteria will be reactivated when the immune system is weakened. So this is known as post-pulmonary infection. So this shows that TB is opportunistic infection, meaning that bacteria will infect our system from time to time. Okay, so in general word, TB not only can affect in your lung, the primary infection is infected your lung, but it also can infect your organ. However, your body system only cannot kill the bacteria one. It only can deactivate the bacteria only. It only can hold off the bacteria inside your lung only, meaning that it only can halang prevent your bacteria not spreading to other part only. But if your immune system is very weak, this bacteria will have the possibility to infect the other organ again. So the primary infection of your organ is your lung. So normally the symptom of this bacteria is causing battle more than 3 weeks, chest pain, fever, night sweating, fatigue, shivering, loss of appetite, breathing difficult, and wheezing. Okay, the transmission of this disease, the transmission vector is A. So it is like similar to COVID-19 one. COVID-19 is also airborne disease, right? And also it affected your lung. But the difference of the infection agent, COVID-19 is virus, but this infection agent is bacteria. So TB is an airborne disease. So normally it can through the droplet infection when the infected person is released its droplet. So the bacteria are spreaded through the air when infected person with TB cough, sneeze, and talk. And then this when there are droplet droplet. So the in the air is already contain TB lah. So the person that near to this kind of person, you will have the high possibility to get the TB lah. So like a COVID-19 lah. So TB is look like the similar to COVID-19 lah. Okay, and needed to spread when we contact with infected person, and only infected person with active TB can transmit the disease. Only this kind of person already infected with TB can transmit the disease. Others cannot. Okay, so there is a high risking of contracting with TB in overcrowded house condition with poor ventilation. So normally if you not do the social distancing, when there are many people are crowded to each other, then you have the high possibility to get TB lah. Okay, this is due to this bacteria can survive in the air and in the house dust for a long time period, and then the people may be diagnosed TB although in the house if they do not clean the house often. Okay, so this is the, as I mentioned before, mycobacterial bovis is that you get this bacteria is come from the cow, right? So normally if you drink the unsterilized milk, you drink the milk that directly come from milk, yeah, you also have the possibility to get this kind of disease. So low protein diet will cause the weaker or immune system, and then cannot violate the bacteria within the lung. So normally the weakened immune system, like the patient, ARDS patient, diabetes patient, or you have cardiovascular, or you are drug abuse, you will very easily to get the TB lah, because your immune system now is very weak, cannot fight against this bacteria. And then like the person who work in hospital, prisoner, penjara, also have the high risk to get. Okay, so this TB actually you can assume that TB is like the COVID-19 lah, because the transmission method is totally similar from the COVID-19. Okay, the prevention. Okay, about the prevention of TB, normally we already nowadays the scientists have already created the vaccine called BCG vaccine, but this BCG vaccine only can cause the death of mycobacterial bovis only. The mycobacterial tuberculosis, no, it cannot kill yet. So normally it only can protect children only, but it does not give much protection in adult. So normally the protection is not suitable lah. It's not suitable for malnourished children as their diet do not have enough protein to make antibody. And then it is important that vaccinate as many as possible to create the herd immunity lah, like COVID-19 lah. Although vaccine is not 100%, but if you 100% can kill the bacteria, but if there are many percentage of the person get the vaccine, you will get the herd immunity to prevent the spreading of TB. And normally this TB patient will be isolated in house, like the quarantine lah, like what you do in COVID-19, you will be quarantined for a period. Okay. And then when the people exposed to TB should be contact tracing. This one I think everyone should know, right? Contact tracing, you need to trace all the people that had already exposed to you. So you will know that there are many trace like in what you happening in COVID-19. Okay, so normally this is the prevention of the TB. Okay, next is about the malaria. Okay, malaria is the infection of the blood that is carried from the person to person by female Anopheles mosquito. So this is another type of the disease that transmitted by the mosquito. So it is like that you learn just now, like dengue lah, but the infection agent is different. The infection agent is protozoa. Protozoa. So you need to know the example lah. So the most famous example is Plasmodium falciparum. So there are also others: Plasmodium vivax, Plasmodium malaria, Plasmodium ovale, and Plasmodium knowlesi. But the Plasmodium falciparum is the most famous one lah. So it is caused by Plasmodium species, and then the transmission vector is female infected mosquito called Anopheles. Okay, so normally it began with a bite of an infected female Anopheles mosquito to the uninfected person, so that it will introduce this protozoa, this infection agent through the Anopheles saliva into your blood circulatory system, the people lah. So in the blood, your protozoa will travel to liver and then mature and reproduce. Okay, so this is the transmission method, very important lah in malaria, the life cycle of the malaria. So as I mentioned before, when the Anopheles mosquito bite you, it will inject the sporozoid into your blood, and then this sporozoid will enter into travel into your liver and infect the liver, and then it will multiply in the liver to form merozoid. So you can see that it will infect your liver and multiply and then to form merozoid. And then once the merozoid will infect your red blood cells. So once they infect the red blood cell, they will develop into the ring form. And then to form the trophozoid and also zytone, that in turn produce further merozoid. So it will repeat again to form more and more merozoid, and then once your red blood cell is already infected by merozoid, the red blood cell will burst and then to release this parasite cells, and then this parasite cell will affected again your red blood cell again. So as a result of this extensive division, medium of this parasite present in the human blood, and then and then it will produce the gametocyte of the protozoa. Okay, once there are another Anopheles mosquito bite you again, you now the infected person bite you again, it will take up this gametocyte. Okay, so this gametocyte is taken again, and then so this mosquito now is already infected with the malaria virus, malaria protozoa. It already contain the sporozoite again. So this mosquito is already infected, and then it will go to another person, uninfected person, transfer this sporozoite again and infected again and again. So Anopheles mosquito is a key transmission vector that affect the human to cause this malaria disease. So the prevention method normally is similar to the prevention method that you in dengue, because you need to prevent the biting of Anopheles mosquito, right? So normally the prevention method is look similar, almost similar. So I think I don't need to explain more. So actually that's all for this chapter. So this chapter is very short and very easy chapter for me. It's very easy chapter lah. So okay, so that's all for today. Okay, that's all for the revision that I already conduct 2 days. Okay, next I will go to the so our last class, so now is just 3 o'clock. So we will continue the last session that I said, which is the analysis of Stpm semester 2 that I said before, and the lesson I said is 4:30, right? Because I end this chapter more earlier, so we will conduct the analysis session in 4 o'clock lah, at 4 o'clock. So we will see you again the final lesson for 4 o'clock, at 4 o'clock. Okay, so we see you again lah. Okay, so thank you. Regarding the slide, for me, I just I plan to sales up draw I plan to sell our slide, my notes. So later I will share the link for those who are interested to buy. You can buy lah. Okay. Okay, so now we take a rest for 45 minutes, and we see you again at 4 o'clock. And then that time is about the analysis lah. And also, if you have any question, you can ask lah at that time. Okay, thank you. So see you again. Okay. Okay. So testing. Okay. Okay. So now it's already 4 p.m. So I will start the analysis now. Okay. So normally the analysis, I make analysis based on the structure question and also the essay. So I only make an analysis for structure part, section B, and also essay part lah. So we will continue to the essay part first, structure part first. So normally, as you guys know, that structure question got 2 questions, right? And then you guys need to answer both questions. Okay, answer both questions. Okay, so actually if you see these later I will share this into the group. Okay, so normally in structure question, the analysis is almost not very in overall process. No, normally there are also many repeated question. So normally, especially if you see this chapter 7, the lung volume already come out two times already, and then if you see the topic about the gestalt human just only come out once only in 2019 about the dissociation curve, and then another one in gestalt implant only come out once in 2021. And then another one hot topic is about the cardiac cycle, especially the cardiac cycle graph. It already come out 2 times too. So 2014 already come out, and then another one is come out in 2019 Ulangan repeat paper also come out, also same as the graph, and then also in 2022 also come out the question about the cardiac cycle, but it is not a graph lah, it is a general cardiac cycle question only. And then if you compare to the disease, so far disease is not come out yet in structure question yet, but it normally can be come out in essay lah. Okay, and lymphatic system so far also not come out yet, but in transpiration process, especially the movement of water implant routes, that is the 3 pathway, vacuolar pathway, simple pathway, and apopass pathway, already come out many times lah. It also come out 2 times if I'm not mistaken. So normally it already come out in 2015 repeat paper, and then one more time is come out in 2020 repeat paper. So come out in 2 times, and both are come out in repeat paper. And then in translocation so far come out once in 2017 repeat paper. So okay, so in general you don't think that the question already come out, even though it is structural question already come out in past year already come out, you don't think that it will not come out yet in future. So yeah, it maybe will come out, especially this kind of topic. This is why I say as a hot topic lah. Okay, and then about the chapter 9, so normally the nerve impulse is not yet come out, but normally it will come out yet. Come out once, if I'm not mistaken, is about the graph lah, 2013, the graph of the action potential. So it only come out once already, so it already more than 10 years not come out already. And then about the nervous system in muscular junction, so normally structure of neuromuscular junction come out in 2015, and then only come out once, and then after that in 2021 the sarcoma of the muscle, basic unit of the muscle tissue sarcoma is come out in 2021. And then another one is about the steroid hormone. Okay, so normally non-steroid hormone is come out in 2023 that you said, right? You said last year, right, about the non-steroid mechanism hormone, and then you see just come out in 2021 repeat paper only. the gap is only 1 year only 1 year, then it come out again. So this one you tell you that don't think that don't think that MPM will come out repeat the question that is very far away from very long times ago. No, it can be come out in only even though 1 year gap. Even though only 1 year gap, it will come out one more times. So for those 2023, for those last year, you maybe you got do the past year 2021 repeat paper, then you are lucky lah. You should can be score lah. If not, yeah, you are not lucky lah. and then you see about the chapter 10 reproduction. So normally sexual reproduction in human menstrual cycle is already come out in 2019 repeat paper. So you don't think that the question that you learn in SPM will not come out in Stpm, because menstrual cycle you already learn in SPM, right? So many people think that I never come out lah, will not come out one lah. But yeah, it will become out, although it is repeat paper, but it also possibility to become out. Okay, and also 2022 repeat paper 2 will come out and already come out about the embryonic development. Okay, another one is flowering plant, sexual reproduction in flowering plant never come out yet in structural question. And then about the seed germination also never come out, but the growth come out. Okay, the growth, one of the famous question is 2014 repeat paper, which require you to draw the growth curve. You want to draw it, will MPM provide you graph paper, and then you need to draw it. So and then before you draw, you need to calculate the growth rate. So there are two types. You know that we have three types of growth curve, right? It come out two types: relative growth rate curve and growth rate curve, and also another absolute growth rate curve. So you need to how to calculate this growth rate, so that you know how to draw. If not, then you don't know how to draw, and then you lose mark lah. Because it already come out, so it may be possibility come out again. Okay. And then one of the famous is hormone in metamorphosis. Hormone in metamorphosis is also very famous question, because it come out 2 times too. So in 2015 repeat paper, and another one is 2018 come out already. Totally, I can say totally similar lah. Totally similar, almost similar. So yeah, you need to alert lah. And then also in 2020 repeat paper, the come out growth in annual plant. Okay, the limited growth, unlimited growth in annual plant. Okay, and then about the homeostasis part, the most famous question is liver, about the blood glucose level, because blood glucose level already come out many times. 2014 one time, and then 2018 repeat, and then 2023, yeah, your last year you said already come out 3 times already, and total almost similar about the negative feedback mechanism of blood glucose, and similar to essay question also repeat many times this kind of question. So you need to know lah, you need to alert. And then amount of also osmotic regulation in mammalian, normally it come out V-adsorption of nephron structure, and also V-adsorption in proximal convoluted tubule come out twice for this topic. For ADH and the adsorption so far no come out yet in structural question, and implant never come out yet in structural question. And immune system, yeah. And the immune system, the most famous question is antibody. Antibody come out twice, 2016 and also 2017 repeat. So you also see I only get 1 year only. 1 year, after 1 year they come out again, but although it is in repeat paper lah. Okay. And then about the development of immunity, it just come out once in 2018 about the cell-mediated immune response. Okay. And then concept cell and non-cell never come out yet. And then about the infection disease, all topic is come out except dengue. Dengue never come out yet in structural question. So maybe possibility to this year come out, maybe. So normally dengue, cholera, TB and malaria already come out, and then malaria it come out twice. So in 2013 repeat paper and also 2020 already come out, but the type of question is differ. 2020 it come out is about the life cycle of the Anopheles mosquito, but in 2013 the repeat paper one is the come out is about the normally is about the general cause, general symptom only. Okay, so this is all about the structural question. So in general, structural question is very difficult to spot because too many things. I just can tell you that some of the topic is considered as hot topic that will maybe come out repeated come out in your exam. If you see your last year paper structural question actually already come out. If you do past year, you should score structural part because non-steroid hormone already come out or come out in 2021 repeat, and also blood glucose already come out twice, already come out at 2018 repeat and also 2014. So last year is very lucky lah. You are very lucky last year because the structure question are totally repeat question. So actually last year structure question is very easy lah, I can say, because totally repeat. The MPM is repeating come out the question. So for last year you should be able to score lah. But unfortunately, maybe you are unable to score, or maybe your essay is worse, so you cannot score in your semester 2, and you need to take repeat. Okay. So for now, because you already lose your chance, so for this year 2023 repeat, is it NPM were like last year, more bad-hearted, more charity to you that you will come out the repeated question again, or it will come out never come out yet question. So we don't know, nobody's know. So for structural question it's very hard to spot. I just want to tell you that no, don't think that the question already come out in past year will not come out in future. So all the topic you should know. So because structural question you don't have to choose, you don't have choice, no choice, you need to answer all. So not like essay you can choose. So yeah. Okay, so this is all about the structure. Next I go to the essay. So essay is the most important lah. So normally essay I can say that due to the semester 2 there are doubling paper compared to Sem 1 and Sem 3, right? Because in semester 2 you have repeat, so normally you have the double past year compared to Sem 1 and Sem 3, because 2013 until 2023 already 11 year, and then you have repeat again at more 10 year, so you are total have 23 question, 23 set question in past year for semester 2. So because of this, so almost all topic in semester 2 are coming out, and then there are many repeating questions starting 2020. Okay, so if you starting in 2020, you can see that the question that I highlight I give the color, normally get color, is the repeating question. It is the repeating question. So you will see the 2020, 20, the repeating question. These are 2020 for the pressure flow hypothesis is the first time that come out in 2020, and then it is repeating again at 2020 repeat paper. Just only 2 year gaps, 2021 and 2022, or not only 2 year, 1 year gap only, because 2021 not come out yet, 2022 in real not come out, but in repeat paper it come out. Totally same about the pressure flow hypothesis, totally same. So yeah, this is one of the example, and then you see in 2020 got another repeat, yeah, and then this one describe the 3 plant growth regulator and their function in agriculture. And their function in agriculture. So this one is also repeated from previous. So you see here you see that there are already plant hormone already come out many times, like the auxin hormone, auxin role, auxin function, acetic acid function, all these already come out. And then even though if you in the future in 2022 and 2022 repeat, yeah, you can see that totally same. Although the hormone is different, but the topic come out is same. Many people will assume that, think that 2022 already cut out plant hormone, 2000 repeat paper tak akan keluar lah. No, MPM keluar lagi. And then starting 2021, MPM always repeat the question. Why I say like that, you will see in 2021 and 2021 repeat, if you refer back 2021, it already come out development of atherosclerosis lead to angina pectoris, already come out atherosclerosis once. In repeat paper it come out again, want you to explain the development of atherosclerosis again. Although the question does not mention about atherosclerosis, but your answer it should be write out the development of atherosclerosis, because they say that developing of lumen could lead to damage of artery, and how is the damage possible? Come out again, totally almost similar. I cannot say totally lah, but almost same, because it's under same topic. Repeat again, because many people think that already come out this topic, so repeat not come out. But so no one want to read again, will focus other topic. But when they get the exam, they found that repeat the question. So rugi, siapa yang rugi? Awak yang rugi. So we don't know what are the mind of the MPM. MPM memang macam ni punya, memang dia boleh main-main dengan awak punya. You don't think that awak jangan anggap yang keluar, even though just in the previously, dia tak akan keluar lagi dekat repeat paper, dia akan keluar lagi. And then in 2031 is also very serious here, because it come out not only first question HIV keluar lagi. Yang ni totally sama. You see 2021, not only the first question about the development atherosclerosis, but ini pun sama. And then I can say this totally sama about the HIV infection. So and then the marks given also same. So totally MPA just copy and paste totally copy and paste the question. So 2021 is the year that MPM normally is doing copy and paste. 2 questions already doing copy and paste. So for those who in 2021 can detect, for those who repeat and then they think that MPM tak akan keluar lah, yang dah keluar kan tak akan keluar lagi lah, so they don't read about HIV and then they come out again, and then people don't read, rugi lah awak. Kerana MPM dah bagi clue lah, bagi markah free bonus kepada awak. But if you think no, you never come out, then rugi lah awak. And then starting 2021 and 2022 also similar, also got 2 question repeat. Why? 2022, one of the repeat question is plant hormone, because 2022 real exam already come up plant hormone, already MSC acid. 2022 repeat still same topic, but the hormone is different. Because some people think that, many people think that, already come up plant hormone, so 2022 repeat will not come out lah, will never come out lah. But PM, what mind awak, they come out again, but just different hormone. So for those who not read the plant hormone topic in 2022 repeat, then you rugi lah, karena dia keluar lagi. And then 2022 also got 1 repeat question in immune response. I can say totally same, just that the difference is the pathogen. 2022 is you need to describe immune response infected by bacteria. 2022 repeat paper is toward cancer cell, but the answer, the general concept is totally same. You need to mention about immune response and also cell mediated immune response. Totally same. 9 marks. So if you think that already come out, never come out lah 2022, but it come out again. Already 2 years, starting 2021. I don't know MPM buat apa. 2021 and 2022 already 2 question, it repeat again for real paper and ulangan. So in 2023, who knows? Siapa tahu? Maybe it will come out again about the maybe come out again control breathing mechanism, maybe come out again, maybe about the somata mechanism. For this one is closing, right? Maybe come out opening again, nobody's know. And then maybe this one, how blood in the human heart is enjoy to flow in correct direction, maybe it come out again about you need to describe the structure of mammalian heart. No, because under same topic, right? Same topic. So nobody's know. And then this one, describe 3 pathway of the water movement. Maybe it repeat again. Nobody's know. And then another one after that is about this one, like dengue. Maybe this year they repeat again dengue. Totally repeat. I can say totally repeat because 2021 HIV is totally repeat, and then your immune as well is totally repeat. Or maybe not using dengue, it come out cholera, come out TB, come out malaria, like that lah. Nobody know, because you don't think that the chapter that already come out in real exam, in repeated exam will not come out. You still have the possibility to come out. Even though in the similar topic have the possibility to come out, same chapter pun ada possibility to come out. So this is a very dangerous lah, because in you refer to past year for infection disease for infection disease, if already come out in real exam, repeat exam will not come out. If come out in real exam, repeat exam will not come out. Come out real exam, but in 2015 come out already break the rules. 2015 come out in real exam, 2015 repeat also come out. And then if you see the MPM in semester 2, dia memang MPM memang main awak punya. Main macam mana? Mesti 2015 20A it come out 20A 20B. If you refer to one of the year, they maybe will come out in different question one. Maybe it come out 20A and then come out 18B. So one of the example later I will give you example. So 2015 and 2015 repeat already put the role. So don't think that the chapter that infection disease, I focus on infection disease. Don't think that infection disease already come out in real exam, repeat will not come out. It still had the possibility to come out. 2016 never come out, not come out. 2017 not come out, but 2018 come out again. Come out again. About the infection disease, repeat not come out. 19 no, 18 no, 20 no, 21 yes, 21 repeat yes, 2022 no, 23 no. 23 is your this dengue, right? So maybe this year repeat it come out another disease, nobody know. So yeah, you need to focus. So the more though I need to focus of you as well is because starting 2021 and 2022, MPM is taking the copy and paste, taking the question from the real exam. So I encourage you, maybe this year MPM will take similar things. So the topic that you come out in 2023, this one, all these dengue, about the dengue, and then what come out in 2023, and also water movement in plant roots, and the blood, human heart, about the human heart, topic in the human heart, and then the topic regarding the breathing mechanism and the stomata. You need to know also. Don't think that come out never come out in repeat, because already 2 years, 2021 2022 MPM do things like this. So maybe 2023 it will come out like this too. So you need to alert lah. Memang kena alert. And then in essay question, essay Sem 2 is the most repeated question compared to Sem 1 and Sem 3. Because Sem 1 and Sem 3, your number of paper is less. Because especially Sem 1 is only containing 20, and you only have 2013 until 2024 only, because no repeat. There are no addition repeat paper special for Sem 1. And in Sem 3 only repeat for 2013 until 2015 only. Starting 2016 no repeat as well. So your number of paper for Sem 1 and Sem 3 is less. But in Sem 2, due to there are the special paper for repeat, so there are many repeat questions. So if we see one by one, for gastroenteritis human, so far no repeat yet. So carbon dioxide transport already come out. Carbon dioxide and also Bohr effect also come out, but our effect also come out, and 2022 repeat oxygen dissociation also come out, but until now never come out yet. Already maybe 2 years, 2 years never come out. So maybe 2023 repeat maybe come out. And breathing mechanism is 2023 is the first time it come out. Never come out yet before. Never come out. You see. Then starting for 2013, it never come out. 2023, your batch is the first time it comes out. So maybe some of you lah, maybe you already refer past year. Now you think that never come out, maybe you not come out lah this year. But MPM keluar lah. Dah keluar lah. So yeah, you cannot play with MPM. Nobody's know. So dia keluar memang keluar. So I can repeat again. Any syllabus in mentioned by MPM memang akan keluar punya. Okay. Next is about the stomata. Okay, stomata it come out many times. Stomata opening come out twice already. 2020 repeat paper come out, and then 2019 repeat paper come out. Only 1 year, 1 gap only. You see that? Totally same mechanism of opening of stomata. Totally same. Only 1 year only no, only 1 year, not 1 year, just 1 semester only. Because many people think never come out lah. Dah keluar kan, takkan keluar lah. But only one gap, come out again. And then not only this, okay, so mata closing also come out in 2013. And then in mammalian transport for the heart, it come out 2019. It already come out, describe the control of heart rhythm. Okay, and then another one is production of first heart sound, cardiac cycle. 2021 is the first year it come out. Cardiac cycle is coming out in essay question. And then 2022 repeat, still the same cardiac cycle. And then 2023 you see under same topic, heart come out again, but the topic is the subtopic is different, but it is under the same topic. And then this I don't need to mention 2021 already repeat, totally same. And then already come out many times 1, 2, and then atherosclerosis 3 times. 4, 2017 come out 4, and then 2013 come out 5. Along these 10 years, it already come out 5 times about the topic atherosclerosis. A very high chance, right? 5 times. 5 kali lah dia keluar. So you don't know this year your year come out or not. And then about the lymphatic system, it come out once in 2013 only. Once only. And then never come out before. Come out once only in 2013, and then never come out before. Come out, never come out. Got 2017. But it come out is about the functionless of lymphatic system. 2017. And then after that never come out yet. Covered before lah until now. Okay, and then about the transpiration, transpiration also the famous topic similar to atherosclerosis, especially transpiration pool and correlation tension theory. You will see here, transpiration pool come out once in 2000 repeat, second 2015 also come out, and then third it also come out, and then in 2018 correlation tension theory and transpiration pool, and then this time also your movement of water in root tissue also already come out in 2018. And then 2020 come out yet, number 4 times. And then you go to here already under topic transpiration pool already come out 4 times. And then in your 2023, this prescribed 3 pathway already come out in 2018. Totally similar. Just the difference is 2018 already mentioned the pathway. You need to explain the pathway. But the 2023 NPM more hati lagi, because dia langsung tak ada bagi pathway. You need to mention. You just mention the pathway, then you get the marks already. Get the marks already. So totally repeat. Yeah. So starting this year, starting 2020, every year containing repeated question. You will see your 2023. You will see your 2023. Which question is repeat? First, 19B already repeat. And then after that is second, dengue. Already got 2 question is repeated. Dengue prevention, because dengue is coming out in 2013. 2013, 10 years. Totally 10 years. After 10 years, it repeat again. So you need to alert. Okay. And then we go to the chapter translocation. I already mentioned, right? In 2020, it is the first time the translocation topic is coming out. And then in 2022 repeat paper, it come out again, repeat again. So just only 2 years gap, it coming out again. And then about the nervous system, in 2000, it is the first time come out in 2017 repeat paper about the action potential and also transmission of nerve along the neuron. And then 2019 it coming out resting potential. So on just 1 year gap it come out again about, but it is resting potential. And then 2020 repeat it out about the influence of speed of conduction of action potential. So okay, so this is the topic under nerve impulse. So far synapse is never come out yet. Synapse never come out yet. So yeah, maybe this year come out. We don't know. So another one is about the neuromuscular tension and muscular contraction. So 2019 is the first year coming out about the transmission, but it does not come in transmission of impulse to the at the neuromuscular junction, but the muscular contraction is not coming out yet, but it coming out first time is 2022 repeat paper, muscle contraction. Okay. And then another one is about drug. Drug it coming out twice about QV already coming out twice in 2017 and also 2015 already come out twice, but cocaine never come out. And the peripheral nervous system with parasympathetic and sympathetic ones never come out yet in essay, so maybe come out. And then the mechanism of hormone, steroid and non-steroid, it just come out once only at 2018 repeat. Come out once only, and then never come out yet. And then this one is just come out differentiation. They never come out want you to explain about the mechanism. They just come out the differences only. They never come out yet about the want you to explain about the mechanism, never come out yet. And then the plant hormone is the famous topic, come out many times in past year. Already come out how many times you see? 2014 come out, both 2014 both come out, 14 and 14 repeat twice already. 2016 come out yet, 2020 come out again, 2022 and 2022 repeat come out 6 times already. This is the very hot topic. Plant hormone is considered as very hot topic. Along these 10 years, 11 years, it already come out 6 times. So the chance is very high. And then you don't think that 2022 already coming out, 2023 not coming out. Nobody know. Nobody know. About the cytochrome action flowering also come out twice. 2020 repeat come out once, and then this year you will see that MPM sangat kejam, because the MPM does not mention about the long day part and short day part. You need to determine which is the short day, which is the long day. So for this, like this, you will see that cyclomenterminus and spinach. So we know that cyclomenterminus is short day, spinach is long day. You need to know. If you don't know or you reverse, then you say that cyclomenterminus is long day, totally salah. 0 marks as well. Memang salah, totally salah. MPM memang kejam punya. Sangat kejam kan MPM? Kejam. So if you make mistake, memang 0 mark. Although your explanation about the cytochrome action is true, but since you already misarrange, you already falsely say that the cyclomentus is long day plant, spinach is short day plant, then totally no marks as well in this question, because in your initial part you already say wrong. Initial part wrong, then totally wrong lah. So you need to know lah. So don't think that, especially in the steroid and non-steroid hormone, it can be also using this like this as example. Maybe you want you to describe the mechanism of acetylcholine. Mechanism of progesterone. You need to know progesterone is what, steroid hormone or non-steroid hormone. You need to know lah. And then not only that, maybe in compare, it will not give the direct question like this. It will maybe differentiate between progesterone and insulin. So you need to know lah. Progesterone is steroid or non-steroid. Insulin is steroid or non-steroid. You need to know. So not just that you just know the concept only, you just also need to know the example. You need to apply them. If not, the question give the example, you don't know how to apply, then rugi lah. And then about the flowering, it come out twice. 2020 repeat and 2018 repeat. And then also 2016 come out 3 times already. But only real agent in 2016, and then it come out again 2018 repeat and come out again in 2020 repeat. And then in sexual reproduction topic, normally it come out is relate to hormone in during pregnancy. You will see here, the first one is come out is placenta, formation of placenta, and also the structure of placenta and their function. And then about hormone, now 2014 repeat come out hormone, and then you see another hormone during in pregnancy. And then 2017 repeat is the, I think is the lucky pair spermatogenesis. Too lucky loh. This is a candidate, I can say you are very lucky lah if you come out this kind of question. Too easy, 9 marks, too easy lah, because you already learn SPM too. So maybe it come out oogenesis also very easy, but you also already learn SPM, so you should know. It come out, you are very lucky lah. And then hormone question, I already say come out many times. First time, second time, and then you see third time. You also hormone in pregnancy, but this one is early pregnancy. Difference from this, 2017 is a long pregnancy, but for this one it's limited to early pregnancy only. So you need to alert. Don't all the hormone you simply hamplan all give up. You all give up, then some of the hormone is not occur in during early pregnancy. For example, like oxytocin oxytocin is only occur at the late of pregnancy. Normally oxytocin is only appear in during the birth. And also PCTH, PCTH also PCTH or ACTH will only appear late of the pregnancy. Okay, next, already come out 3 times about the hormone during pregnancy. So you need to alert lah. And then about the sexual reproduction in flowering plant, so far because this topic only have 2 topic, double fertilization and embryonic development, right? So embryonic development already come out twice, 2015 and 2022 come out twice. All totally similar. Embryonic development, right? Totally similar. And then another seed germination also come out many times. 2022 you see the 2022 repeat question already. 2 questions are repeated. So this year who do a past year, you are very lucky lah. If you already do a past year, this year candidate, you already do a past year, you are very lucky lah. Because 19 marks, you should have 19 marks at your hand now. Because you already do past year, you should know. You refer to the MPN laporan, you should know. Why? Because you see seed germination 2022, and then 2016 repeat come out one more time. 2014 come out. Already 3 already. You already have 2 example question. So you should know. And then embryonic development you already come out 2015. So 2022 candidate are very lucky lah. You are very lucky lah. But unfortunately you are not 2022, you are 2023. Okay. And another one, growth, yeah, also come out many times. Okay, it come out first time is 2016. It want to explain about the growth pattern of human and fish, and about the metamorphosis of insect. Come out once already. And then you will see that metamorphosis 2016 come out, and then it come out again 2020. Wow, repeated again and repeated again. 2020 it come out again. So, and then not only that, growth also come out. Growth pattern of human and fish come out, and then growth pattern of crab also come out, and the growth curve also come out. So you see that 2017 and 2017 repeat similar topic. Just the one is growth curve, one is growth pattern. So you don't repeat again. You don't think that in real exam already come out growth. Repeat will not come out growth. Come out again. So, and 2017, I think this one is very critical because nobody knows growth pattern of grab. What is this? So yeah. So, and then 2020, it come out limited growth and unlimited growth, about the differences. Okay, and then about the homeostasis. So importance of homeostasis, it come out once in 2015, repeat. And then another one is come out in 2018, about the why that we need to regulate our body temperature and also blood osmotic pressure. Why we need to regulate it. Okay, and then the most important is liver, about the carbohydrate metabolism and lipid and protein metabolism. How many times you see 2017, yeah, similar again. 2017, totally repeat question, you will refer to it. 20A already mentioned about the blood glucose. He want you to write down how the liver can return the increase the blood glucose level to normal. And then you will see this, when the blood glucose level is decreased, how do you return normal? Totally same, almost same, not totally, 100% same, copy and paste. So 2017 already break these rules already. And then starting 2021, MPM break the rules again. And then 2022 break the rules again. So 2023, nobody's know. So, and then already come out, carbohydrate metabolism come out twice. And then you refer back again, yeah, mechanism regulate human blood glucose, third. And then you will see again, third time, already come out 3 times already. And then about the amino acid, come out first time in 2017, and then second time come out in 2019, repeat. And then another one 2020, repeat again. So you see, repeat, already come out 3 times. Okay, and then you see and then osmoregulation in mammalian, or this one, although it come out many times, and then but every time the question is not repeated. Not repeated. So you see in 2016 it already come out, is about the role of kidney in osmoregulation. So this is about the ADH hormone. You need to explain about the ADH hormone. And then another one is 2017 repeat about the reabsorption. And then 2018 is about the ADH hormone again, but the condition is different. You see, because in 2015 repeat, it say that it is just the general situation. After the athlete is do a complete long distance marathon, what are the role of kidney? But for this one, 2018, is that why we can still survive even though we take a low amount of water. So although it is still similar to ADH, but the answer right is quite different. The concept is still same, but the keyword, the terms that you use, you need to add on some similar terms. You can refer to MPM report. Okay. 2019 formation of glomerular nitrate, so ultrafiltration already come out, and then homeostatic mechanism of sodium ion when a person is lost a block of blood. So this is the first time your aldosterone renin enzyme system is coming out, and then it come out again about the ADH, but the question is changed a lot because it say that when your posterior pituitary gland that plays secret ADH is malfunction, cannot function, or already cannot function improperly, what happened? Okay, what happened? Okay, so this osmoregulation, remember, it can come out in K part 1. Although the concepts used is similar, but since it is some of the K part questions, so your answer need to change some based on the question. You cannot write the question in very similar, very general concept. If you write in general concept, then you still have a marks, but you cannot get full marks. Okay. And then osmoregulation imply, this one is also very famous. About the halophyte and xerophyte, right? When it come out how many times? First time 2013 repeat, second time 2014 repeat, third time 2016, fourth time 2020, fifth time 2022. 5 times already come out. You see, 5 times. So it is also one of the hot topic, and this topic nobody want to read. Yeah, nobody want to read, but they normally will skip out this question. So up to you. You want to skip, up to you. But normally this question will be similar to the osmoregulation one. So you see 2022, 20B is about this, but your 20A is this. Wow, embody development. You know how to write. But 20B, you don't read. 5 marks gone. And then you see this 20B. Wow, 20B 9 marks, but 28 which what question? 28 you see is this about the plant growth regulator. Wow, and the function in agriculture. Yeah, you got read, but you see 20B. Alamak, why this? I never read. Rugi lah. And then you will see again 2026, 2016. Oh, you don't read 19B. Okay, 19A is what? 19A is this. Oh, easy lah. I got read, I know. But 19B, no, I don't know because I don't read. So how do you answer? And then this is the part lah. I can say it is very tricky because if you see that 19A and 20B, although it is under same topic 10.4, but MPN separate into 2 different questions. It actually, if MPN by RT, it can be in the same question one. It can be 19A is describe the growth pattern, and 19B can be describe the metamorphosis one. But MPN do what? MPN na mind awak, they want play with you. So they are separate it. So lah, by 19A I know how to answer, but 19B, hello, apa ni? I don't know lah, I never read. Rugi lah. Macam mana awak nak dapat markah? And then you will see that 20B, you know how to answer, you read it. But 20A is what you will see, refer back 20A. 20A is 20A. Ah, 28 is this. So 28 is you can answer lah. So for this year, you rugi lah. Memang saya tak tahu macam mana bantu awak lah. Kerana 19A you know how to answer 8 marks, but 7 marks you don't know. And then same 2014 repeat. 2014 repeat is more MPM more hati lah, kerana dia terus satu soalan sekaligus. So if you don't know, then you don't need to answer it. And then about the immune system, chapter 12. So it also come out many times. 2013 repeat. Explain the role, especially this. Explain the role of lymphocyte in immune system already come out 3 times. How many? 3 times. 2013 repeat. And then you see that 2019 repeat, come out already, second time. 2021 repeat, 3 times are coming out in repeat. Real exam never come out yet, but repeat come out. So maybe 2023 repeat come out. We don't know. I don't know. You will know it next Wednesday. Then you know. So yeah, you need to alert. And then about the lymphatic system come out twice. 2021 repeat, and also 2014 repeat. So you see, refer that immune system all are coming out in repeat one, except 2019 only. 2019 it come out if it want you to classify the 5 class of antibody. Wow, too easy lah. They say too easy lah. Okay. And then this one 2014 repeat is one of the many people cannot answer it, especially 2020B. Because in your book, normally if you not refer to, not even though in Campbell or Solomon never mentioned about the lymphoid organ much, so you need to refer others book, especially you need to refer immunology book, then you will know how to answer it. If not, you cannot answer it. Okay, about the development or immunity. Okay, this one actually in the beginning of the Stpm never come out yet. It come out first time is 2016 repeat paper, and then it come out again 2018 repeat paper, but it come out in repeat paper, and then it come out again in 2019 repeat paper again, and then 2021 is the first time it come out in real paper, real examination, and then 2022 I don't know why MPM come out copy and paste. Dia tak keluar banyak kali, tapi dia keluar, and dia keluar copy and paste. So 2022 already come out, and 2022 repeat also come out, and then 2022 already repeat already, because 2022 is repeated from 2019. 2019 already come out once, 2022 already come out once, and then 2022 copy and paste again to 2022 repeat. Wah, MPN tahun ni memang malas tau. Dia malas lah, baby, dia malas keluar soalan lah. So yeah, you need to alert lah. About concept cell and non-cell, on this topic also many people will not answer it, especially organ transplant. But HIV most of you answer it lah. So HIV already come out 2013 repeat, and then it come out again in 2021, and 2021 dia copy and paste. Wah. So no, you don't know MPM dia fikiran dia buat apa punya. Nobody know. Yeah. And then immune disorder, many people will not read this, but it will maybe will possible come out. It come out once in 2018 repeat, 5 marks only lah. Okay, and then infection disease. This one I need to focus, because meaning of infection disease come out many times. You see 2021 repeat come out. What is mean by infection agent and infection disease? And then 2018 come out again, 2015 come out again 3 times already, 2014 come out again 4 times. And then structure question also possible come out. If you're not mistaken, 2020 2021 or 2022, if not mistaken. 2022 repeat structural question already come out 5 times about the definition of infectious disease. Already come out 5 times. Wow. So you need to alert lah. So maybe this year repeat come out again. Yeah, lucky lah. You are very lucky lah. Because normally it come out will give you 3 marks. Maximum is 3 marks. And then dengue already come out twice, 2020 and also your batch 2023. And cholera also come out once in 2014, 2015. Yeah, but after that never come out yet. So maybe this year come out. Your repeat paper come out. And then TB only come out in repeat paper. 2021 repeat about the transmission, and then 2015 repeat about also transmission. Repeat again. Just that 2015 it add on the prevention so TB although it come out but in real exam never come out yet. So yeah, maybe possibility you come out. Malaria is only come out once at 2018 only. Come out once only. 2018 only. About the transmission too. And then 12 marks. So in conclusion, I can conclude that since 2021 and 2022, 2 years and directly 2 years, MPM doing copy and paste. So 2023 has the higher possibility MPM doing copy and paste again. So the topic that come out in 2023, I think your paper, your semester 2 biology paper never come out, never throw into the rubbish yet, I think. So if you're not throwing your rubbish yet, please refer back to your paper again, do it again, and then do it again. Don't think that the paper that already come out last batch, your last year, never come out in next Wednesday. So you need to alert this. Okay, so that's all for the analysis for semester 2, and then I just want to share with you the topic that never come out in semester 2 yet. So normally because of the semester 2 have the doubling paper compared to sem 1 and sem 3, so almost all topic already come out I can say compared to sem 1 and sem 3. Because sem 1 sem 3 due to the lack of paper, so in sem 1 sem 3 there are many topics never come out compared to semester 2. So semester 2 actually there are many topics already come out So normally if you see this chapter, only the first topic, gastroenterology and human, only these 3 subtopic never come out yet. About the human respiratory system, structure of the alveolus, and also structure of hemoglobin and transport of oxygen in blood. These 3 subtopic never come out yet. For these 2, breathing mechanism and the gastroenterology impact already come out And then about the heart, mammalian heart structure never come out yet, and the regulation of heartbeat is never come out yet. Initiation is already come out. Regulation of heartbeat is never come out yet. And then disease, I think I don't need to say already come out many times. And lymphatic system already come out, but many years never come out already. Transpiration also already come out many times, but translocation, mass flow hypothesis already come out, but another 3 hypotheses is never come out yet. Electro-osmosis, cytoplasmic streaming, and peristolic wave hypothesis is never come out yet. So maybe come out this year, maybe no. So and then about the chapter 9, nerve impulse is about the organization of nervous system in human is never come out yet. And then the structure of sinus and its transmission is also not come out yet. Others already come out. And then structure of numerous muscular junction no come out yet. And then the parasympathetic and sympathetic comparison never come out yet. And then the cocaine drug mechanism also not come out yet. And then the steroid or non-steroid is already coming out lah once. And then plant hormone and cytokine action I don't need to mention lah, because already come out many times. Especially plant hormone already come out 6 times. The most hot topic. And then cytokine action already come out twice. And then about the sexual reproduction, oogenesis, fertilization, role of hormone in menstrual cycle, stage in embryonic development, and role of chorion and meiotic fluid and allantoin in fetal development never come out yet. Others already come out. And then sexual reproduction in flowering plant, double fertilization never come out. And then seed germination all already come out lah. Growth curve and pattern of growth already come out, and including metamorphosis already come out. And then homeostasis topic, important homeostasis already come out. Thermoregulation also already come out. Liver, I don't need to mention lah. Carbohydrate metabolism, liver metabolism all already come out. And then about the osmoregulation in mammalian, normally formation of UV just includes secretion never come out yet. Ultrafiltration and reabsorption already come out. And then regulation of pH never come out. And then osmoregulation in plant, this topic, although many people don't want to read, but it come out many times. And immunity is about the development of B cell and T cell are never come out yet, because it is very hard topic. This one you need to read a lot baru our understand, if not you cannot understand. So and then about the development of immunity, the most famous one, cell mediated immune response and humoral immune response already come out. Just that complement fixation never come out yet. Okay, concept self and non-self organ transplant already come out. HIV already come out. Immune disorder already come out once in 2018. And then infectious disease all are come out already come out past year. So you will refer back. Actually, the topic that never come out is very less. So this is also the topic maybe will be come out in 2023. Repeat. Here, this is your. So I waiting your paper lah next Wednesday. Okay, so that's all for the analysis. So I think that's all for today lah. Today class, and that's all for this 2-day class. So final, I wish you, I hope that this 2-day class, that those who attend, you can get the final revision. You know what I am teaching, you know what you need to learn, you already master all the semester 2 syllabus if you want to get A lah. Okay, so hopefully all of you want to get A, right? So final word is about good luck for your repeat paper, ulangan 2, ulangan 2. Semoga anda boleh berjaya lah. Next week I think already, right? 15, right? 15 April you were starting to start your repeat paper, right? 15 April or 16. Or 16 April you will start repeat, but your biology is 17, right? So not only biology, others repeat paper like chemistry, PA or MAT, you if you are repeat, good luck for your U2. And then after U2, congrats, you are graduate in your Stpm lah. So final word, good luck for your examination. And then take your time these few days to take a break and take a revision, final revision for your ulangan 2. And then that's all for this 2-day revision class. Thank you for those who are attending, and then good luck for your exam. So thank you, and then have a nice day, and have a happy Raya, and then happy holiday after your U2. Okay, thank you and goodbye.